Good evening, and welcome to The Sleepy Scientist. Tonight's episode is a calm journey into the chemistry that makes life possible, biochemistry. Whether you're tucked beneath a blanket, listening on a quiet commute, or just looking for something peaceful before bed, this is your time to slow down and let your mind wander through the invisible machinery of life. Before we begin, I'd love to know, where in the world are you listening from tonight? It's always fascinating to see the different places these calm stories drift out to. If you enjoy these gentle explorations of science, a like and a subscribe help the channel more than you'd think. And to everyone who's already subscribed, thank you for being part of this growing little corner of calm curiosity. Don't forget to tap the notification bell so you don't miss the next upload. I do hope the video isn't too interesting that it keeps you awake. But if it does, at least you'll fall asleep a little wiser about the chemistry quietly keeping you alive. So what exactly happens inside every cell of your body when molecules meet, react, and somehow create life? Biochemistry is the science of living chemistry, a story of how the same handful of atoms that form rocks and air somehow learn to think, move, and dream. It's the quiet explanation behind every heartbeat, every breath, every spark of thought. At its heart, biochemistry is built on three ideas. First, structure and function. The shape of a molecule determines what it can do. Second, energy flow. Every living thing must take energy from its surroundings and use it carefully. And third, regulation. The dance of chemistry must be controlled, or life quickly dissolves back into chaos. Water is the stage upon which all of this unfolds, and energy is the thread that ties it together. Molecules like proteins, lipids, and nucleic acids are the actors, each performing small, precise roles within the vast play of metabolism, and through it all the same rules of physics and chemistry apply. Life doesn't break the laws of nature, it simply bends them elegantly. This video takes a slow walk through those ideas. We'll explore how cells use enzymes to guide reactions, how energy moves from sunlight to sugar to muscle, and how information written in DNA becomes action. We'll visit the molecules that make you, not just as a collection of chemicals, but as a living pattern that renews itself every moment of your life. No background in chemistry is needed here. Think of this as a story told under soft light, a look into the clockwork of biology, ticking quietly while you rest. So settle in, slow your breathing, and let's begin our journey into the quiet chemistry of life, where atoms learn to live. And just before we begin, this video is for educational and relaxation purposes only. It is not medical advice and shouldn't replace guidance from a qualified healthcare professional. Biochemistry is the quiet meeting point between chemistry and life. It asks one simple question that unfolds into countless others. How do lifeless atoms come together to create something that eats, breathes, and dreams? Every living thing, from a single bacterium drifting in a puddle to the mind listening to this sentence, is built on the same small cast of chemical players. Carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur form nearly everything alive. Yet when these elements connect in precise ways, they gain something remarkable: organization, renewal, and purpose. At its simplest, biochemistry studies the molecules that make up life and how they behave inside cells. It's the science of reactions happening quietly in the background, enzymes guiding transformations, molecules passing messages, and energy flowing from one chemical form to another. But more than that, it's the study of emergence, how billions of random molecular motions come together to create something steady and meaningful. To understand it, it helps to think in layers of scale. At the smallest level are atoms, the building blocks of everything. When atoms link up, they form small molecules like water, carbon dioxide, or glucose. These in turn join to form macromolecules, large complex structures like proteins, DNA, and membranes. Step up another level, and those macromolecules organize into pathways, chains of reactions that turn raw ingredients into useful products. And finally, those pathways form the foundation of cells, the smallest self-contained units of life. A cell, seen under a microscope, looks calm, but at the molecular level it's a storm of reactions. Tens of thousands of different processes happen every second, each one balanced with the next. It's not chaos, though. It's choreography, a constant push and pull that keeps the cell alive and stable. This idea is key. Life is not a single thing, but a state of dynamic balance. The molecules that make you are constantly changing, but the pattern they form remains the same. You are, in essence, a moving equilibrium, never still, never the same twice. Biochemists sometimes describe this balance with the term steady state. Unlike a rock which sits at equilibrium and does nothing, living systems resist settling down. They burn fuel to stay slightly out of balance, holding themselves in a delicate tension that allows work to be done. Energy is taken in, used and released again, like a gentle tide flowing through every cell. When that flow stops, life stops too. To make sense of all this activity, biochemists often use what's called the central dogma, the flow of information from DNA to RNA to protein. It's the idea that DNA stores instructions, RNA carries those instructions, and proteins carry them out. It's not a rigid law, more of a guiding principle, a way of understanding how information turns into action. DNA tells a cell what to build. Proteins do the building. Together they form the quiet loop that allows life to copy itself and continue. But it's worth remembering that DNA is not a dictator barking orders from the nucleus. It's more like a library, silent until someone opens a book. The environment, the cell's needs, and countless small chemical signals decide which pages are read and when. In that sense, life is a conversation between information and circumstance, not a script read word for word. When you zoom out, the beauty of biochemistry lies in how familiar rules produce unfamiliar results. Atoms obey the same physical laws inside a cell as they do in a rock, yet only in cells do those laws create movement and meaning. The difference is organization. A living system is an open system, one that exchanges matter and energy with its surroundings. It's constantly taking in nutrients, releasing waste and managing gradients, differences in concentration or energy that allow work to happen. Gradients are life's quiet engines. They're the reason your nerves can fire, your muscles can contract, and your cells can absorb nutrients. A cell builds and maintains these gradients, spending energy to separate what's inside from what's outside. Like a battery charged across a membrane, the imbalance itself becomes useful. Through countless tiny gates and pumps, ions and molecules drift, flow, or are carried from one side to the other. The balance between order and disorder, input and output, defines what it means to be alive. And in the middle of all that activity sits the same repeating pattern that will echo through everything you'll encounter in biochemistry: structure, function, and regulation. The structure of a molecule determines what it can do. A protein's shape decides which reactions it can guide. Its function is the job it performs, whether that's carrying oxygen, breaking down sugar, or copying DNA. And regulation is how the cell makes sure that job happens only when needed, never too fast or too slow. This trio, structure, function, regulation, keeps cells from dissolving into chemical mayhem. It's what makes biochemistry both predictable and endlessly surprising. You can know every atom in a cell and still be astonished by how they behave together. The next time you look at your hands or take a breath, remember, everything happening inside you is chemistry, but not the kind found in beakers and test tubes. It's a living chemistry, constantly adjusting, constantly renewing itself. If you could shrink down to the size of a molecule and float through a single human cell, you'd find yourself surrounded by shimmering networks of proteins, spirals of DNA twisting and untwisting, and thousands of chemical exchanges happening faster than thought. Yet amid all that activity, there's a sense of order, a kind of quiet cooperation. Cells are like quiet kitchens. The recipes are precise, the timing is flawless, and the timers never beep. Every molecule knows its role, every reaction its cue, and together they form something far greater than the sum of their parts, the chemistry of life itself. Water is the quiet foundation of life, the gentle stage upon which every molecule performs. Without it, biochemistry would grind to a halt. It is easy to overlook because it is everywhere, but that's precisely what makes it extraordinary. Water isn't just a background ingredient, it's an active participant in nearly every process that defines being alive. A single water molecule is simple, two tiny hydrogen atoms joined to one larger oxygen atom, forming a shape that resembles the ears of a small invisible mouse. But that slight bend, that asymmetry, is what gives water its remarkable character. The oxygen holds electrons a little more tightly than the hydrogens, creating a separation of charge, one side slightly negative, the other slightly positive. This polarity turns every water molecule into a tiny magnet, endlessly attracted to others like it. Because of this, water forms hydrogen bonds, fleeting attractions between the slightly positive hydrogen of one molecule and the slightly negative oxygen of another. They're weak individually, breaking and reforming billions of times per second, but together they create a delicate yet resilient network. This invisible lattice is what gives water its unique personality, its high surface tension, its ability to dissolve so many substances, and its stubborn resistance to temperature change. These traits aren't trivial quirks. They're the reason your body doesn't freeze solid on a cold night or boil over on a warm day. Water absorbs heat without changing temperature much, creating stability in an otherwise unpredictable world. Oceans hold warmth through the night, cells maintain their internal balance, and organisms can thrive across extremes of environment. That same network of hydrogen bonds makes water the perfect solvent, often called the universal solvent, though that's poetic exaggeration. Water's polarity allows it to surround and separate ions and polar molecules, pulling them gently apart and keeping them suspended in solution. When salt dissolves, for example, water molecules swarm each sodium and chloride ion, forming tiny hydration shells that stop them from reuniting. In the cell, this same property allows nutrients, waste products, and signaling molecules to travel freely, like commuters through a transparent city. But water's power to dissolve isn't universal. Some substances, like fats and oils, prefer to stay apart from it. This is the hydrophobic effect, water's quiet insistence that non-polar molecules keep to themselves. It's not that water actively pushes them away, rather, it prefers to maintain its own network of hydrogen bonds. When oily molecules force water to reorganize around them, it becomes energetically costly, so those molecules huddle together to minimize disruption. This simple principle sculpts life's architecture. Cell membranes form because hydrophobic tails of lipids gather inward, hiding from water, while their hydrophilic heads face outward to interact with it. Proteins fold into intricate shapes for the same reason. Hydrophobic amino acids tucked safely inside, hydrophilic ones exposed to the watery world outside. Every shape, every structure in biology owes something to water's quiet preferences. If structure is shaped by water, then stability is maintained by another of its quirks, its relationship with protons. Water can both give and take them. existing as a delicate equilibrium between hydrogen ions and hydroxide ions. When there are more free hydrogen ions, the solution is acidic. When there are fewer, it's basic. The concentration of these protons is measured using a scale called pH, the potential of hydrogen. pH may sound intimidating, but it's just a way of counting how many free protons are in a solution using a logarithmic scale. A pH of 7 is neutral, meaning that hydrogen ions and hydroxide ions are balanced. A pH of 6 means 10 times more hydrogen ions than neutral. A pH of 5 means 100 times more. It's like measuring sound in decibels. Each step represents a power of 10. This compressed scale allows us to describe enormous differences in acidity with small, tidy numbers. Inside your body, keeping pH within a narrow range is critical. Most enzymes, the molecular workers that power biochemistry, are extremely sensitive to pH. A slight shift can change their shape and with it their ability to function. For instance, the enzymes in your stomach are adapted to an environment with a pH around 2, while those in your blood prefer something close to 7.4. Too acidic or too basic, and the delicate hydrogen bonds that hold proteins together start to unravel. To prevent chaos, living systems use buffers, chemical pairs that can absorb or release protons as needed, keeping pH steady even when acids or bases are introduced. Think of them as the shock absorbers of the chemical world. The bicarbonate buffer system in your blood is a perfect example. Carbon dioxide from your cells dissolves in water to form carbonic acid, which can then break apart into bicarbonate and protons. By shifting between these forms, the system can mop up excess acid, or release it when things become too basic. The phosphate buffer system plays a similar role inside cells, balancing local changes that occur as metabolism hums along. Together, these systems create an invisible safety net, ensuring that the inner ocean of your body stays calm no matter what's happening outside. Without them, something as simple as a sprint, a meal, or even a deep breath could throw your chemistry dangerously off balance. This control isn't just theoretical, it's the quiet difference between health and harm. When blood pH drifts too low, a condition called acidosis, enzymes slow, oxygen transport falters, and fatigue sets in. When it rises too high, known as alkalosis, nerves misfire and muscles cramp. Yet under normal circumstances, the body maintains pH with astonishing precision, drifting only by a few tenths of a unit, a feat of regulation so fine-tuned it rivals any laboratory instrument. You can see the beauty of this balance in something as ordinary as breathing. When you exhale, you're releasing carbon dioxide, the same molecule that drives the bicarbonate buffer. Holding your breath traps it, increasing acidity slightly. Exhaling reduces it, shifting the balance back toward base. Even the simple rhythm of respiration is a form of biochemical tuning, a conversation between lungs, blood and brainstem to keep that delicate equilibrium intact. Enzyme activity follows the same principle. Each enzyme has an optimal pH where its shape and charge distribution are perfectly aligned for work. Shift too far, and the charged side chains that hold its structure repel or attract in new ways, warping the active site and slowing the reaction. It's a reminder that even the smallest chemical details, a stray proton here, a shift in charge there, can ripple upward into the larger story of life. So much of biochemistry depends on water quietly doing its job, dissolving what should be dissolved, nudging together what should meet, and buffering against the extremes that would otherwise bring everything crashing down. It's a modest molecule, but its humility hides power. Imagine every droplet in your body, from the moisture in your eyes to the fluid inside your cells, constantly managing an orchestra of charges, bonds, and balances. Each moment, water molecules are rearranging, forming and breaking connections 1 trillion times per second, yet maintaining an overall calm. If chemistry had a heartbeat, it would sound like the gentle rhythm of water, endlessly moving, endlessly adjusting, keeping the fragile machinery of life running smooth. And that, perhaps, is why biochemists so often speak of water not as a solvent, but as a companion to life itself, a partner that never stops working, even when you're fast asleep. Every reaction that happens in your body, from breaking down a meal to forming a memory, follows the same quiet rule. It must make energetic sense. Life, after all, runs on the laws of thermodynamics, not the roaring kind that launch rockets, but the gentle kind that shuffle molecules into new arrangements. Even in the calmest cell, billions of chemical reactions are weighing their options, asking the same question: is this worth the energy? To understand that question, biochemists use a concept called free energy, often represented by the symbol delta G. It's the bookkeeper of thermodynamics, quietly noting whether a reaction releases energy or requires it. A negative delta G means the reaction can happen spontaneously. Energy is released, and the universe gives a small approving nod. A positive delta G means the reaction needs a push. It's uphill, energetically speaking. But spontaneous doesn't mean fast. A lump of iron rusts spontaneously, but you could watch it for hours before anything noticeable happens. Speed belongs to kinetics, not thermodynamics. Free energy only tells you whether a reaction could happen, not how quickly it will. In that sense, delta G is less of a commander and more of a quiet whisperer, a guide that shows which direction the chemical wind is blowing. This whispering balance governs everything from metabolism to muscle movement. For example, when glucose burns inside your cells, it releases a generous amount of energy, giving carbon dioxide and water as byproducts. The delta G for that reaction is hugely negative, so favourable that if it happened all at once, you'd burst into flames. Thankfully, the cell channels it through many tiny regulated steps, capturing energy bit by bit rather than letting it go in a single fiery sigh. Free energy changes come in two main flavours: exergonic, which release energy, and endergonic, which require it. You can think of them like downhill and uphill journeys. Left alone, systems roll downhill toward lower free energy. But life can't survive just by sliding down slopes. It needs to climb as well, to build, organize, and repair. To do that, cells have mastered the art of coupling, linking an uphill reaction to a downhill one so that the energy released from one drives the other forward. The classic example of this partnership is adenosine triphosphate, ATP. Breaking one of ATP's high-energy phosphate bonds is an exergonic reaction. It releases energy. A cell can take that energy and pair it with an endergonic reaction, such as building a protein or moving an ion across a membrane. The two reactions together share a single fate, one paying the energetic bill, the other spending it. It's a bit like a seesaw. On one side, you have a process that wants to go down, on the other, one that needs to go up. Couple them together, and you get a balanced motion. Energy flows smoothly from one side to the other. The cell never wastes energy. It trades it, recycles it, and uses it efficiently in 1,000 quiet exchanges. Behind this dance of energy lies another concept that often gets an unfair reputation: entropy. It's usually described as disorder, but that word misses the point. Entropy isn't about mess, it's about possibilities. The more ways a system can arrange itself, the higher its entropy. Imagine shuffling a deck of cards. When it's freshly ordered, there's only one arrangement like it. After shuffling, there are countless combinations, each one just as likely. Entropy has increased not because things are messy, but because there are more available states. In biology, entropy isn't an enemy to be defeated. It's the quiet partner that ensures energy flows in a direction. Every spontaneous reaction increases the total entropy of the universe, even if a cell locally creates order. When you build muscle or copy DNA, you're not breaking the second law of thermodynamics. You're just borrowing energy from the environment to pay entropy's fee. Life survives by constantly spending energy to keep itself organized while allowing the universe as a whole to grow more disordered. It's a tidy compromise, and it works beautifully. The relationship between free energy and entropy is neatly summarized in a single equation. Delta G equals delta H minus temperature times delta S. In plain language, that means the change in free energy equals the change in enthalpy, or total energy, minus the temperature multiplied by the change in entropy. You don't need to memorize it, but it's comforting to know that even life's complexity can be written in a sentence of mathematics. The balance between energy content and freedom of motion determines whether something will happen. In the laboratory, scientists measure these reactions under standard conditions, a fixed temperature, pressure, and concentration. But life doesn't live in a lab flask. Inside cells, conditions are anything but standard. Concentrations fluctuate, temperatures vary slightly, and molecules are rarely left alone. That's why biochemists often talk about actual free energy, the delta G that applies inside living systems, not the idealized one on paper. A reaction that seems unfavorable in theory can become possible in the cell, where concentrations are carefully tuned and reactions are coupled together in intricate networks. This brings us to one of life's most elegant tricks, maintaining a steady state. In chemistry, equilibrium is when the forward and backward reactions balance perfectly, and nothing changes overall. In a living system, reaching equilibrium would be disastrous. It would mean no more energy flow, no more work, no more life. Instead, cells maintain a steady state, where the concentrations of molecules remain stable, but the reactions never stop. It's like water flowing through a fountain. The shape stays the same, but the water itself is always moving. That constant flow requires energy, of course, but it allows the cell to control direction and speed. By adjusting enzyme levels, energy supply, or the concentration of key molecules, the cell can shift the balance, nudging reactions gently toward where they're needed. It's a kind of thermodynamic diplomacy, maintaining peace among countless competing reactions. Nowhere is this more beautifully displayed than in the flow of energy through metabolism. Energy captured from sunlight or food is transformed into chemical bonds, then passed through molecules like NADH or FADH2 before finally reaching ATP. Later, ATP's energy is spent to drive pumps, synthesize molecules, or send signals. It's a continuous loop, a currency system that never sleeps. Even the quiet act of breathing is a thermodynamic exchange. Oxygen accepts electrons at the end of a long chain of reactions, allowing cells to extract energy efficiently from fuel. The byproducts, carbon dioxide and water, are released, and the process begins again. Every breath you take, every heartbeat, is powered by tiny shifts in free energy, measured in thousandths of a joule per molecule, but multiplied across trillions of reactions. Thermodynamics, then, isn't just a set of equations. It's the hidden rhythm of life. It decides which reactions proceed and which are postponed, guiding the chemistry of existence with an invisible hand. If you were to imagine life without these rules, it would be like a game with no scoreboard, moves made without consequence, energy spent without purpose. But with them, every reaction has meaning. Every molecule knows its place in the grand flow of energy that began billions of years ago, when sunlight first touched the ocean and simple chemistry began to stir. So the next time you feel the warmth of your skin, or hear the soft exhale of your breath, remember that you are, in a sense, a collection of thermodynamic conversations, each molecule whispering its preference, each reaction finding its balance. Life doesn't fight the laws of physics, it listens to them very carefully. And in that quiet obedience lies the secret to everything alive. If thermodynamics decides whether a reaction can happen, kinetics decides when. A reaction might be favourable on paper, but without a little help, it could take longer than the lifetime of the universe. That's where biochemistry's unsung heroes come in. The enzymes, catalysts of life, speeding up reactions that would otherwise crawl at a glacial pace. Every chemical reaction faces an invisible barrier called activation energy. Even when the end result is more stable, the molecules first need to twist, stretch, or partially break bonds before new ones can form. It's like rolling a ball from one valley to another over a small hill. Thermodynamics tells you the ball wants to end up in the lower valley, but kinetics tells you how long it will take to climb the hill. Enzymes lower that hill, making the journey faster and smoother without changing the final destination. They're not consumed in the process, they simply make it easier for reactants to find each other and react in the right way. The result is breathtaking efficiency. Some reactions that would take centuries in plain water finish in fractions of a second inside a cell. Enzymes are the world's politest speeders, fast but never reckless. They accelerate chemistry with precision, guiding molecules as if traffic lights and roundabouts existed at the molecular level. Each enzyme is a protein folded into an intricate three-dimensional shape, and that shape is everything. Hidden within it is a small region called the active site, where the magic happens. The active site is like a snug pocket, perfectly shaped to hold the molecule or substrate that needs to react. But it's not a rigid mold. Early scientists once imagined enzymes as locks and substrates as keys, a neat but slightly outdated metaphor. Today we understand it more as an induced fit. When the substrate enters, the enzyme subtly shifts its shape to hold it better, wrapping around it in a brief and perfect embrace. It's less a lock and key and more a handshake that adjusts naturally to the other person's grip. This flexibility allows enzymes to stabilize the unstable transition state, that fleeting moment between old bonds breaking and new ones forming. By lowering the energy required to reach that point, enzymes make reactions proceed thousands, sometimes millions of times faster. Each enzyme has exquisite specificity. It recognizes its substrates not just by shape, but by charge, polarity, and even subtle patterns of hydrogen bonding. The tiniest change, a flipped bond, a missing atom, can turn a perfect fit into a complete mismatch. This is why a single mutation in DNA can sometimes alter enzyme behavior dramatically. Change one amino acid in the active site, and suddenly a reaction that once hummed along smoothly grinds to a halt. To describe how enzymes behave, biochemists use a model known as Michaelis-Menten kinetics, a mathematical way to explain how reaction rates change with substrate concentration. You can imagine it as a traffic analogy. When there are only a few cars on the road, enzymes can easily handle the flow, each car representing a substrate molecule quickly finding an open lane. As traffic builds up, more cars compete for limited enzyme sites. Eventually every enzyme is busy, fully saturated, and the reaction speed plateaus. That plateau is known as the maximum velocity, or Vmax. The point at which the reaction rate is half of that maximum is defined by a constant called Km, the Michaelis constant. Km tells you how much substrate is needed for an enzyme to work at half its full speed. A small Km means the enzyme binds tightly to its substrate. Even a little substrate makes it run fast. A large Km means the enzyme needs more substrate to get going. These simple ideas, Vmax and Km, let scientists describe enzyme efficiency without peering directly into the microscopic whirlwind of reactions. In the living cell, enzymes rarely work at their maximum speed. They are tuned to balance the needs of the moment, adjusting through temperature, pH, and regulation by other molecules. It's the biochemical equivalent of a conductor holding back a full orchestra, ensuring every instrument plays at just the right volume. Enzymes can also be slowed down or stopped entirely by inhibitors, molecules that interfere with their activity. This isn't always a bad thing. In fact, it's how many medicines work. The simplest form is competitive inhibition, where another molecule mimics the substrate closely enough to compete for the active site. It's like someone sitting in your favorite chair before you can. The real substrate can't bind until the imposter leaves. Many drugs use this trick deliberately. For example, statins lower cholesterol by competing with the natural substrate of an enzyme involved in cholesterol synthesis, reducing its output. Another subtler form is non-competitive inhibition. Here, the inhibitor binds to a different part of the enzyme, changing its shape just enough to affect the active site indirectly. It doesn't block the doorway. It simply warps the frame. Even if the substrate can still enter, the enzyme no longer performs its task properly. This form of inhibition can't be overcome by adding more substrate, because it alters the enzyme's very behavior. In the body, such inhibition is often used for regulation, allowing feedback loops to keep reactions from running wild. There's also uncompetitive inhibition, a rarer type where the inhibitor binds only to the enzyme-substrate complex, locking it into a kind of frozen embrace. The reaction can't complete, and the product isn't released until conditions change. It's like pressing pause on a molecular movie. These forms of control allow life to fine-tune its chemistry with astonishing precision. Imagine if every enzyme in your cells ran at full speed, all the time. You'd burn through your energy stores in seconds. Instead, enzymes respond to the cell's needs, ramping up when energy is required and slowing down when balance returns. One of the most fascinating things about enzymes is their sheer diversity. There are enzymes that break things apart, hydrolases, enzymes that build new bonds, ligases, enzymes that shuffle groups around, transferases, and enzymes that change shapes, or isomers, isomerases. Each has a name that sounds like a gentle incantation from a chemistry textbook, but together they manage nearly every process of life. Digestion, DNA replication, nerve signaling, even the way you think and move. They're also surprisingly resilient. Some enzymes survive boiling temperatures in deep-sea vents. Others operate near freezing in Arctic fish. There are even extremophiles, whose enzymes function perfectly in acid strong enough to dissolve metal. Each one has evolved to balance flexibility with stability, just loose enough to move, but sturdy enough to hold shape. At the molecular scale, it's all a game of probabilities. Substrate molecules bounce around, colliding billions of times per second. Most collisions are misses, wrong angle, wrong timing, but enzymes make success almost inevitable. They pull molecules into the right orientation, stabilize fleeting intermediates, and release products with quiet satisfaction before starting all over again. Despite their elegance, enzymes aren't flawless machines. They make mistakes, misfire, or occasionally latch onto unintended partners. Yet even their errors can be useful. Evolution itself thrives on tiny imperfections. A slightly altered enzyme might gain a new ability, opening doors to new pathways or adaptations. Over billions of years, this molecular tinkering has written the recipe book of life. When you zoom out, enzyme kinetics tells a comforting story. Life doesn't need brute force to work. It runs on subtle guidance, a steady rhythm of controlled acceleration and careful restraint. Every heartbeat, every breath, every thought relies on enzymes quietly ensuring that the chemistry of living stays just fast enough and never too fast. So the next time you digest a meal, flex a muscle, or simply take a slow breath, remember that inside you are trillions of microscopic speeders, polite, tireless, and perfectly timed. They don't shout or rush. They simply make the impossible happen at a pace the universe can live with. If enzymes are the engines of life, then regulation is the steering wheel. Without control, metabolism would be a noisy, chaotic traffic jam, reactions racing ahead with no coordination. But inside every cell, there's remarkable order. Reactions speed up, slow down, or switch direction, as if following an invisible conductor. The secret behind that harmony lies in how enzymes are regulated. A mix of elegant self-control, long-distance signaling, and molecular common sense. One of the most graceful forms of regulation is allostry, from the Greek words meaning other site. Allosteric enzymes have more than one binding site, and what happens at one site affects what happens at another. Imagine an enzyme as a flexible sculpture that can subtly shift shape. When a small molecule, called an allosteric effector, binds to one part of it, the enzyme's shape changes slightly, often far away from where the main reaction takes place. This shape shift can either turn the enzyme on or off, depending on what the cell needs. The result is a beautifully simple feedback system. If a product starts to build up, it can loop back and bind to an earlier enzyme, telling it to slow down production. This is known as feedback inhibition, and it's one of biology's favorite tricks. For example, when a cell has plenty of the amino acid isoleucine, that very molecule binds to the first enzyme in its own synthesis pathway and gently tells it to rest. No shouting, no alarms, just a polite molecular whisper that says, "We have enough, thank you." Allostry also allows enzymes to act as sensitive switches. Hemoglobin, though not an enzyme, is a famous example. It changes shape as oxygen binds, making it easier for the next oxygen molecule to join, a phenomenon called cooperative binding. This principle, borrowed by countless enzymes, ensures they can respond dynamically to tiny shifts in concentration. It's as if the cell can turn dials with infinite precision rather than flipping crude on-off switches. While allostry handles many short-term adjustments, some enzymes need longer-lasting control. That's where covalent modification comes in. This means attaching a small chemical group to an enzyme to change its behavior, often by adding or removing a phosphate group in a process called phosphorylation. It's the cell's equivalent of toggling between active and standby. Phosphorylation is fast, reversible, and remarkably effective. A single phosphate group carries a strong negative charge, and attaching it to a protein can shift the protein's shape, alter its charge distribution, or change how it interacts with other molecules. Enzymes called kinases perform the attachment, while phosphatase remove the phosphate when it's time to turn things off again. Together, they create an instant messaging system for cellular chemistry. This form of control appears everywhere, in metabolism, in cell signaling, even in memory formation in the brain. It's astonishing to think that something as simple as a phosphate group can decide whether a muscle contracts or relaxes, whether a neuron fires, or whether a cell divides. But cells have another layer of regulation that's more about when and where an enzyme acts than whether it's on or off. Some enzymes are made in an inactive form, called zymogens or proenzymes, and are only activated when needed. It's a clever safety feature, especially when the enzymes involved are destructive by nature. Digestive enzymes are the classic example. Trypsin, which breaks down proteins in the small intestine, is produced by the pancreas as an inactive precursor called trypsinogen. Only when it reaches the intestine is it safely converted into its active form. This prevents it from digesting the very tissues that made it. It's the biochemical equivalent of keeping sharp knives in their sheaths until you're ready to use them. Compartmentalization serves a similar purpose. Cells are not just tiny sacs of liquid. They're divided into carefully managed regions, each with its own tasks and conditions. By keeping certain enzymes confined to specific compartments, like mitochondria, lysosomes, or peroxisomes, the cell prevents cross-reactions and maintains order. It's a bit like having separate kitchens for baking, brewing, and chemistry experiments. Compartmentalization also allows cells to maintain different environments side by side. One area might be more acidic, another more oxidizing, depending on what reactions need to occur there. It's astonishingly efficient, and when combined with enzyme regulation, it creates layers of control that make life both robust and adaptable. Sometimes enzymes don't even wait for molecules to find them. They come together in assemblies called multi-enzyme complexes. These clusters keep sequential reactions close together, allowing intermediates to pass directly from one active site to the next without drifting away. This process, known as metabolic channeling, prevents side reactions and speeds everything up. You can picture it as an assembly line in a calm, silent factory. Each worker knows exactly when to receive a part and when to pass it along. Instead of individual reactions happening randomly across the cell, everything happens in a smooth sequence. The fatty acid synthase complex, for instance, assembles long chains of carbon atoms through a series of reactions, all coordinated within one enormous molecular machine. Metabolic channeling also reduces waste. If intermediates are unstable or reactive, keeping them confined ensures they don't escape or harm the cell. It's a subtle kind of protection, efficiency, and safety rolled into one. At the broadest level, enzyme activity is also guided by signals that come from far away, from hormones that travel through the bloodstream carrying instructions. Hormones are like chemical messengers announcing changes in the environment. Food has arrived, or energy is low, or prepare for rest. When insulin is released after a meal, it binds to receptors on the surface of cells, triggering a cascade of phosphorylation events inside. Enzymes that promote energy storage are activated, while those that release energy are quieted. Hours later, when blood sugar begins to drop, another hormone, glucagon, delivers the opposite message, switching the system back again. These hormonal controls turn individual enzyme reactions into an orchestra, playing in perfect time with the body's needs. From stress responses to sleep cycles, hormones provide a unifying rhythm that keeps thousands of enzymes working toward the same goal. Taken together, these mechanisms of enzyme regulation form an exquisite network of control. There's feedback inhibition, ensuring no pathway runs wild. There's allostery, allowing enzymes to sense and respond to their surroundings. There's covalent modification for quick toggles, zymogens for safety, compartmentalization for order, and hormones for long-range coordination. It's tempting to think of the cell as a busy factory, but that's too mechanical. It's more like a living ecosystem, enzymes adapting moment by moment, whispering to each other through molecules, balancing energy and order. There are no foremen shouting commands, no central clock, just chemistry, self-regulating and harmonious. In the end, enzyme regulation is what turns chemistry into life. It ensures that reactions happen not only at the right rate, but in the right place and time, every time you blink, digest, breathe or think. These controls are at work, maintaining a quiet order within chaos. Without them, the cell would be like a symphony with every instrument playing at once. But with them, the music of metabolism flows, complex, responsive, and beautifully composed. If life had a currency, it wouldn't be gold or paper or even electrons. It would be a small molecule called adenosine triphosphate, or ATP. Every cell in every living thing, from a bacterium to a blue whale, uses ATP as its daily allowance of energy. It is not dramatic, not volatile, and certainly not explosive, despite what many textbooks used to imply. Instead, ATP is a steady, reliable middleman, the quiet accountant of life's energy transactions. ATP is built from three main parts: a nitrogen-containing base called adenine, a 5-carbon sugar called ribose, and a chain of 3 phosphate groups extending like a tail. It's that tail that gives ATP its remarkable properties. The bonds between the phosphates are often described as high energy, which conjures images of tiny molecular firecrackers waiting to burst. But that phrase is a bit misleading. The bond itself doesn't contain stored energy like a coiled spring. The key lies in what happens when the bond breaks and water steps in to rearrange things. When ATP loses one of its phosphate groups, it becomes ADP, adenosine diphosphate, and in the process, a phosphate group is released. The reaction is called hydrolysis, literally splitting by water. The energy released doesn't come from the bond breaking alone, but from the difference in stability between ATP and its products. A DP and the free phosphate are more stable in water because their negative charges can spread out and interact favorably with their surroundings. It's this shift, this move from a tense, crowded arrangement to a calmer, more relaxed one, that releases usable energy. So ATP doesn't explode, it exhales. It's not a battery that dies with a spark, but more like a rechargeable adapter that connects one form of energy to another. That adaptability is why life chose ATP as its universal energy carrier. Cells use ATP to power nearly everything that requires energy. Muscle contractions, nerve impulses, protein synthesis, active transport, and even the subtle flicker of molecular repair. Yet it's never hoarded. The human body, for example, contains only about 50 grams of ATP at any given moment, barely enough to power a few minutes of life. But that same ATP pool is recycled endlessly, broken down and rebuilt over and over. In a single day, your cells turn over their entire stock of ATP thousands of times. Each molecule is spent, recharged, and spent again in a continuous loop that keeps you alive. One of the reasons ATP is so versatile lies in its ability to transfer phosphoryl groups, those small, negatively charged phosphate clusters that can attach to other molecules. This process, called phosphorylation, changes the chemical personality of the receiving molecule. Add a phosphate, and suddenly a molecule becomes more reactive, more soluble, or better able to participate in a reaction. Cells harness this transfer in two main ways. The first is substrate-level phosphorylation, which happens directly during reactions like those in glycolysis, the breakdown of glucose. In this case, an enzyme transfers a phosphate group from one molecule straight onto ADP, forming ATP. It's simple, direct, and effective, but it produces only a small amount of ATP overall. The second method is more elaborate and far more productive, oxidative phosphorylation, which takes place in the mitochondria, the cell's energy factories. Here, electrons from food molecules travel through a chain of proteins embedded in a membrane. As they move, they drive protons across the membrane, creating a gradient, a kind of stored potential energy. When those protons flow back through an enzyme called ATP synthase, the enzyme uses that flow to join ADP and phosphate together. It's like a molecular water wheel turning in reverse, crafting ATP from the invisible current of life. Both methods, though different in scale, serve the same purpose: keeping the cell stocked with enough ATP to fuel whatever needs doing next. But why did life settle on ATP in particular, rather than some other molecule? After all, chemistry is full of compounds capable of releasing energy. The answer lies in a subtle balance of kinetics, stability, and accessibility. ATP is stable enough that it doesn't break apart spontaneously in water, yet reactive enough that enzymes can harness its energy on command. It sits comfortably in the middle of the energy scale, not too strong, not too weak, which allows it to couple easily with countless reactions. If it released too much energy, it would be dangerous. If too little, it wouldn't be worth the effort. Another important factor is magnesium, the unsung partner of ATP. In the cell, ATP rarely exists alone. It's almost always bound to a magnesium ion. Magnesium helps stabilize the negative charges on ATP's phosphate groups, preventing them from repelling each other too strongly. It also helps enzymes recognize ATP's shape correctly. Without magnesium, ATP would be too unruly to handle. In fact, many enzymes that use ATP don't bind to ATP itself, but to this magnesium ATP complex. Cells also carefully regulate the ratio of ATP to ADP and AMP, adenosine monophosphate. These ratios act as energy sensors. A high ATP to ADP ratio tells the cell it's well supplied, so energy-producing pathways slow down. When the ratio drops, it signals that energy is running low, and pathways that make ATP speed up. It's an automatic feedback system that ensures energy is always available but never wasted. In this way, ATP is both a currency and a signal, a kind of molecular language that lets the cell communicate its energetic state. A drop in ATP doesn't just mean we're out of fuel, it triggers entire cascades of responses designed to restore balance. Metabolic pathways adjust, ion pumps recalibrate, and the cell quietly finds its footing again. The beauty of ATP's system is that it's universal. The same molecule that powers human thought also drives bacterial motion, plant growth, and the beating of a hummingbird's wings. It's a shared language written in chemistry, spoken across billions of years of evolution. Even viruses, those minimalist passengers of biology, rely on the ATP of their host cells to copy themselves. Life may differ in form, but energetically, it speaks with one voice. You can imagine the cell as a small city where ATP flows like electricity through invisible wires. Every process, from the smallest enzyme reaction to the grand movements of muscles and neurons, depends on this current. The production of ATP in mitochondria is like a slow sunrise, electrons moving, gradients forming, and energy being passed along carefully to the molecules that will use it. Nothing is wasted. Nothing is left uncontrolled. Despite its central role, ATP isn't the only energy carrier in town. Other molecules like GTP, UTP, and creatine phosphate have specialized duties, but ATP remains the master currency because it connects them all. It's exchangeable, reliable, and accepted everywhere in the biochemical marketplace. The next time you move a finger, take a breath, or blink, trillions of ATP molecules are being hydrolyzed in perfect coordination. Tiny coins of energy spent in an instant, only to be minted anew. It's easy to take for granted, but this constant exchange is what keeps you warm, thinking, and alive. There's something poetic in the way ATP operates. It doesn't demand attention or create sparks. It simply transfers energy from where it's abundant to where it's needed, one reaction at a time. It's the molecular equivalent of quiet generosity, steady, dependable, and invisible. So while the textbooks may call its bonds high energy, ATP isn't a stick of dynamite waiting to go off. It's more like a rechargeable adapter that fits every socket in the cell, always ready to connect, always ready to serve. And in the calm hum of that endless exchange, life keeps going, quietly spending its currency, one molecule at a time. If you could shrink down to the size of a molecule and drift through a living cell, it would feel like gliding through a vast glowing city. Streets of reactions stretch out in every direction, bustling with movement. Some lanes are packed with molecules being broken down, others hum with construction as new compounds take shape. There are intersections, roundabouts, and express routes connecting distant neighborhoods. This is metabolism, not a single pathway, but an intricate map of thousands, all linked together by the steady flow of molecules and energy. Each reaction in this biochemical city is like a small workshop. On its own, it does very little. But when strung together in series, these reactions form pathways, long, purposeful routes that transform one molecule into another. Glycolysis, the gastric acid cycle, and the synthesis of amino acids or fatty acids are just a few examples. Together they form the highways and side streets of cellular life. In this map, molecules travel as if they were citizens commuting to work, raw materials being processed and recycled. The rate at which they move, the pace of the chemical traffic, is known as metabolic flux. Flux is the quiet heartbeat of metabolism, describing how quickly substances flow through a pathway. Too fast, and the cell wastes energy or builds up unwanted products. Too slow, and it starves. Keeping the traffic moving smoothly without gridlocks or accidents is one of the cell's greatest feats. At first glance, the idea of metabolism might seem chaotic. Countless reactions running at once, in every corner of the cell. But this complexity hides an elegant order. Every molecule knows where it's going because enzymes guide each step with precision. Each enzyme acts like a traffic light, controlling how quickly its reaction proceeds. These tiny regulators respond to signals, the equivalent of road signs and weather conditions, adjusting the pace to match the cell's needs. Metabolism is usually divided into two grand districts, catabolism and anabolism. Catabolism is the side of the city devoted to breaking things down, burning fuel to release energy. It's the demolition crew that dismantles larger molecules like glucose, fats, and proteins into smaller pieces. The energy released in these reactions doesn't vanish. It's captured in molecules like ATP or NADH, ready to be used elsewhere. Anabolism, on the other hand, is the construction department, building complex molecules from simpler ones. Proteins, lipids, and nucleic acids all owe their existence to anabolic pathways. These processes require energy, often provided by the very ATP and NADH produced by catabolism. It's a self-sustaining economy. What one half tears down, the other half rebuilds. The true genius of metabolism lies in how these two halves communicate. They're not opposites locked in battle, but partners in a delicate dance. When catabolic pathways run fast, energy levels rise, and anabolic pathways slow down to conserve resources. When energy runs low, catabolism speeds up to replenish it. It's like a city where the power grid automatically balances itself. Lights never flicker, and traffic never fully stops. Certain molecules act as key intersections or hubs connecting many roads. Pyruvate is one of the busiest. It's produced at the end of glycolysis and can go in several directions depending on what the cell needs. With oxygen available, it's converted into acetyl-CoA and enters the citric acid cycle for further breakdown. In low oxygen conditions, it can be turned into lactate to keep energy flowing. Or, if the cell has enough energy, pyruvate can be diverted into building amino acids or glucose again. It's a flexible crossroad, redirecting traffic as conditions change. Acetyl-CoA, the molecule that receives pyruvate's baton, is another vital hub. It sits at the center of metabolism, linking carbohydrate, fat, and protein breakdown to energy production. When acetyl-CoA enters the citric acid cycle, its carbons are slowly released as carbon dioxide, and its electrons are passed along to generate ATP. But acetyl-CoA can also serve as a building block for fatty acids, cholesterol, and other molecules, a reminder that even destruction feeds creation. If you were to look at a full metabolic map, and scientists have drawn them, sprawling webs that look like subway diagrams gone wild, you might wonder how cells keep everything from colliding. The answer is coordination. Reactions are not isolated. They talk to each other through shared intermediates and chemical signals. When one pathway speeds up, another might slow down to prevent overload. When the demand for energy increases, say during exercise, pathways that break down glucose and fatty acids accelerate, while those that build storage molecules take a back seat. This coordination relies on feedback and sensing. Cells monitor the levels of ATP, ADP, NADH, and other key molecules as if they were gauges on a dashboard. A high ATP concentration tells the cell it has plenty of energy, so catabolic pathways ease off. Low ATP or rising ADP levels trigger the opposite effect. It's a constant conversation, a balancing act between making and spending, charging and discharging. What's remarkable is how stable yet responsive this balance remains. Even when your energy demand changes, when you sprint, eat, sleep or think, your cells adjust in moments. Blood sugar stays steady, body temperature remains constant, and energy keeps flowing without interruption. This dynamic equilibrium is called homeostasis, and it's one of life's quiet miracles. It's not about staying perfectly still, but about staying within a range that allows freedom to move. Imagine standing in the surf. The water rises and falls, but you adjust your stance to stay upright. That's what cells do. They don't freeze their conditions, they flow with them. If something pushes metabolism too far one way, sensors respond to bring it back. If energy demand suddenly spikes, enzymes switch on to meet the need. Stability doesn't mean rigidity, it means flexibility with discipline. Within this balance, small molecules often play multiple roles. Citrate, for instance, isn't just a step in the citric acid cycle, it also signals that there's plenty of energy available. When citrate builds up, it tells the cell to slow glycolysis and start storing fat. AMP, on the other hand, acts as a cry for help, a low-energy alarm that triggers enzymes to produce more ATP. These molecules are both participants and messengers, blending chemistry with communication. Even at this scale, metabolism feels alive. It's not just a series of chemical reactions, but a conversation happening constantly, between pathways, between compartments, between signals of abundance and need. The flux of molecules isn't random, it's choreographed, responding to the faintest whisper of change. If you could listen to the sound of metabolism, it wouldn't be noise or chaos, it would be a steady hum, millions of reactions unfolding in sync, turning food into energy, energy into structure, and structure back into fuel. Every moment your cells are navigating their molecular city, rerouting traffic, fixing blockages, and managing resources with quiet precision. And yet, despite all this movement, you never feel the rush. The city never sleeps, but it never panics. It simply flows, stable, adaptable, endlessly alive. If metabolism were a map, glycolysis would be its main street, the pathway that almost every living cell, from bacteria to humans, relies on to make its first supply of energy. It's the biochemical equivalent of a steady jog, reliable, rhythmic, and always ready, no matter the conditions. Before oxygen, before mitochondria, before complex life itself, there was glycolysis, a process so ancient that it's shared across nearly every form of life on Earth. At its core, glycolysis is the gentle dismantling of glucose, a simple sugar, into smaller pieces, releasing a modest but essential amount of energy. The word itself means sugar splitting, and that's exactly what it does. It takes one molecule of glucose and turns it into 2 molecules of pyruvate. Along the way, it earns a small profit of energy in the form of ATP and gathers some high-energy electrons carried by NADH. But glycolysis isn't just a straight road, it's a 2-part journey, an investment phase and a payoff phase. In the investment phase, the cell spends energy to prepare glucose for the reactions to come. It's a bit like priming a pump. You give up some ATP at first to ensure a better return later. In the payoff phase, that investment pays back with interest, producing more ATP than was spent. The net result: 2 ATP molecules of pure profit and 2 NADH molecules ready to deliver electrons for future use. The first step of glycolysis is handled by an enzyme called hexokinase, which attaches a phosphate group to glucose, turning it into glucose 6-phosphate. This step is important for two reasons. First, it prevents glucose from slipping back out of the cell. Once phosphorylated, it's trapped inside, committed to the pathway. Second, the addition of the phosphate primes it for the chemical transformations ahead. In liver cells, a gentler version of this enzyme called glucokinase takes on the same task but with a different personality. Hexokinase works quickly, even at low glucose levels, keeping energy production steady, while glucokinase is more patient. It waits until glucose levels are high, such as after a meal, and then stores the excess as glycogen. The next few steps rearrange and activate glucose in subtle ways, until the molecule reaches a crucial checkpoint controlled by phosphofructokinase 1, often shortened to PFK1. This enzyme is the gatekeeper of glycolysis, deciding whether the pathway should speed ahead or slow down. PFK1 adds another phosphate group to the molecule, creating fructose 1,6-bisphosphate, a form that can now be split cleanly in two. PFK1 is tightly regulated by the cell's energy status. When ATP levels are high, it senses that the cell has enough energy and slows down. When ATP is low and AMP levels rise, it accelerates, telling the pathway to generate more fuel. It's a fine-tuned control system, a molecular thermostat keeping energy in perfect balance. Once the molecule is split into 2 3-carbon fragments, the payoff phase begins. These fragments are gradually oxidized and rearranged, producing energy in the process. By the time the sequence reaches its final step, each fragment has become pyruvate, the end product of glycolysis and one of the most versatile molecules in metabolism. The last step, catalyzed by pyruvate kinase, delivers the grand finale, a final burst of ATP production. Like hexokinase and PFK1 before it, pyruvate kinase is another key control point. It responds to the cell's needs, slowing down when energy is plentiful and speeding up when demand increases. Together, these three enzymes, hexokinase, PFK1, and pyruvate kinase, form the regulatory trio that keeps glycolysis responsive yet stable. When you add everything up, glycolysis yields 4 ATP molecules in the payoff phase, but since 2 were spent earlier, the net gain is 2. It's not a massive profit, but it's reliable, quick, and available anywhere in the cell, even when oxygen is scarce. For many organisms, and for your own cells in certain situations, that reliability is priceless. Once glycolysis is complete, the cell faces a decision: what to do with the pyruvate. Under normal oxygen-rich conditions, pyruvate is transported into the mitochondria, where it's converted into acetyl-CoA, the entry ticket to the citric acid cycle. From there, its carbons are gradually released as carbon dioxide, while its electrons drive the production of large amounts of ATP through oxidative phosphorylation. But when oxygen runs low, during a sprint for example, or in tissues with limited blood supply, the cell has to take a different route. In the absence of oxygen, the electrons carried by NADH can't be passed along the usual chain of reactions in the mitochondria. If NADH builds up, glycolysis grinds to a halt, because the cell runs out of NAD+, the molecule that accepts electrons during the process. To avoid this, cells temporarily convert pyruvate into lactate, regenerating NAD+, and allowing glycolysis to continue. This alternative route, known as lactic acid fermentation, doesn't make extra ATP, but it buys time. Muscles rely on it during short bursts of intense activity, those final seconds of a sprint, or the last few reps of a heavy lift, when oxygen delivery can't keep up with demand. The build-up of lactate can cause a burning sensation, but contrary to old gym lore, it isn't responsible for long-term soreness. In fact, As oxygen returns, lactate is converted back into pyruvate and reused for energy. The system is temporary, efficient, and entirely self-correcting. Some organisms have perfected anaerobic life, using fermentation as their primary mode of energy production. Yeast, for instance, turns pyruvate into ethanol and carbon dioxide, a process that has quietly powered human civilization through bread, beer, and wine for thousands of years. In both humans and microbes, the principle is the same. When oxygen is limited, glycolysis becomes the lifeline, a minimalist engine that keeps energy flowing in the dark. What makes glycolysis remarkable isn't just its age or simplicity, but its adaptability. It's a universal fallback, always on standby, ready to step in when more complex systems can't. It can work fast under pressure, yet it's flexible enough to feed into countless other pathways. The intermediates produced along the way can be siphoned off to make amino acids, nucleotides, or lipids. Glycolysis is not a dead-end road, but a bustling intersection connecting metabolism's many routes. This is one of the reasons it's so tightly regulated. If glycolysis ran unchecked, it would waste valuable glucose or flood the cell with unwanted products. By controlling key enzymes like PFK1 and pyruvate kinase, cells ensure that energy is produced in just the right amount. The moment energy demand increases, when you start running, thinking hard, or even shivering, glycolysis responds almost instantly. It's a biochemical reflex honed over billions of years. Despite its efficiency, glycolysis produces only a small portion of the energy that glucose contains, but its true strength lies in accessibility. It doesn't require oxygen, specialized organelles, or elaborate machinery. It's fast, flexible, and reliable, the equivalent of a well-worn footpath in the cellular landscape, one that's never closed, no matter the conditions. In the grand story of metabolism, glycolysis is the opening act, humble, steady, and indispensable. Its chemistry may be simple, but its impact is profound. Every heartbeat, every thought, every movement begins here, with the gentle splitting of a sugar molecule and the quiet release of life's first sparks of energy. So next time you catch your breath after a sprint or feel the warmth of a meal spreading through your body, you're feeling glycolysis in action, an ancient, tireless rhythm, still keeping time after billions of years. At the end of glycolysis, the cell stands at a crossroads. Each molecule of glucose has been split into two smaller pieces of pyruvate, and now those pyruvate molecules wait for a decision. If oxygen is scarce, they can turn into lactate, keeping the energy flowing for a short while. But when oxygen is plentiful, when the cell can afford to run the full machinery of respiration, pyruvate is invited into the mitochondrion, the cell's quiet power plant. There, it enters one of biochemistry's most elegant circuits, the tricarboxylic acid cycle, often called the TCA cycle or Krebs cycle. Before pyruvate can join the cycle, it must first pass through a gate, a carefully guarded entry point controlled by a huge enzyme complex known as pyruvate dehydrogenase. This complex acts as the customs officer of metabolism, inspecting each pyruvate molecule and converting it into a form the cycle can use. It's not a simple handoff. During this reaction, one of pyruvate's 3 carbon atoms is released as carbon dioxide, while the remaining 2 form acetyl-CoA, a small molecule attached to a coenzyme that carries the reactive acetyl group like a ticket into the next stage of metabolism. This step is irreversible. Once pyruvate is turned into acetyl-CoA, there's no easy way back. The cell commits to using it for energy. The enzyme's efficiency depends on a small but essential helper molecule, thiamine pyrophosphate, derived from vitamin B1. Without it, pyruvate can't be processed properly, leading to an energy shortfall in tissues that rely heavily on aerobic metabolism like the brain. It's one of those quiet reminders that vitamins, though tiny in quantity, play irreplaceable roles in the chemistry of life. Once inside the mitochondrion, acetyl-CoA enters the TCA cycle, a circular series of reactions that extract energy from carbon-based fuels. Unlike glycolysis, which runs in a straight line, this pathway is a loop, a cycle that begins and ends with the same molecule, ready to spin endlessly as long as fuel is available. It serves as a central hub, linking the metabolism of carbohydrates, fats, and proteins. No matter what you eat, sugar, oil, or steak, all roads eventually lead here. The first step of the cycle is a meeting of molecules. Acetyl-CoA, carrying its 2-carbon group, merges with a 4-carbon molecule called oxaloacetate. The enzyme citrate synthase catalyzes the reaction, forming a 6-carbon molecule called citrate. This reaction sets the tone for the entire cycle. Smooth, spontaneous, and highly controlled. Citrate, the same molecule that gives lemons their sourness, now begins its quiet transformation. In the next stage, citrate is rearranged into a socitrate, preparing it for a key step catalyzed by socitrate dehydrogenase. Here, another carbon atom is released as carbon dioxide, and electrons are captured by the molecule NAD+, forming NADH, one of the cell's main energy carriers. Each NADH molecule holds high-energy electrons that will later be used to power the production of ATP. A few steps later, alpha-ketoglutarate dehydrogenase performs a very similar trick, removing yet another carbon atom as carbon dioxide and generating another NADH. The resemblance to pyruvate dehydrogenase is no coincidence. The two enzyme complexes are cousins, both relying on the same cofactors, including thiamine. By this point, the original acetyl group has been stripped down, its carbons released into the air as carbon dioxide. What remains of its energy has been captured in the form of reduced cofactors, mainly NADH and a smaller amount of FADH2. From there, the cycle continues through a series of quieter rearrangements. Succinyl-CoA becomes succinate, releasing a small burst of energy that produces one molecule of GTP, which can easily be converted into ATP. Succinate then donates more electrons to FAD, forming FADH2, before being converted into fumarate and then malate. Finally, malate is oxidized back to oxaloacetate, producing another NADH and closing the loop. The cycle is ready to begin again, each turn processing one acetyl-CoA and yielding 3 NADH, 1 FADH2 and 1 GTP, modest per spin, but multiplied across billions of cycles per second, it becomes the steady pulse of life. What's fascinating about the TCA cycle is that it's not just about burning fuel, it's also a crossroads for biosynthesis, a roundabout where molecules can exit and enter as needed. Intermediates like citrate, alpha-ketoglutarate, and oxaloacetate are constantly borrowed by the cell to build amino acids, fatty acids, or other vital compounds. When that happens, the cell must replenish them through a process called anaplerosis, literally filling up again. One common anaplerotic reaction uses the enzyme pyruvate carboxylase, which takes pyruvate and adds a carbon dioxide molecule to regenerate oxaloacetate. This ensures the cycle never runs out of intermediates, even when they're being siphoned off elsewhere. The TCA cycle is therefore both a source and a supplier, generating energy while feeding the rest of metabolism with the materials it needs. Regulation keeps this complex network in perfect tune. The cycle speeds up when the cell's energy reserves are low and slows down when they're full. NADH, ATP, and other high-energy molecules act as gentle brakes, telling the enzymes to relax when enough energy has been made. When levels of ADP or NAD+ rise, those brakes are released, and the machinery spins faster again. It's an automatic balancing act that prevents waste while ensuring constant supply. It's also worth noting the elegant bookkeeping happening at every turn. For each acetyl-CoA that enters, 2 carbon atoms are released as carbon dioxide, yet these aren't the exact same carbons that arrived. The carbons from acetyl coenzyme A are temporarily stored in the cycle before being released in later turns. The cell doesn't lose track of them, it just recycles them through a complex exchange. At the same time, the electrons stripped from those carbons are handed off to NAD+ and FAD, creating NADH and FADH2, tiny molecular couriers that carry their energy to the electron transport chain. There, in a process that unfolds across the mitochondrial membrane, these electron carriers will fuel the synthesis of large amounts of ATP. Each NADH contributes enough energy to produce roughly 3 ATP molecules, and each FADH to about 2. Multiply that by the vast number of cycles running in every cell, and you begin to grasp the sheer power of this system. A constant, silent conversion of carbon and oxygen into usable energy. Because it's so central, the TCA cycle must also be flexible. When nutrients are scarce, it slows down to conserve. When new fuel arrives, it accelerates. In some tissues, like muscle, it shifts toward maximum energy extraction. In others, like the liver, it lends its intermediates to build glucose or amino acids. The same reactions, different priorities, an orchestra playing different pieces from the same sheet of notes. It's easy to see why the TCA cycle is often called the hub of metabolism. Everything flows through it. Carbohydrates, fats, and proteins all feed into acetyl-CoA before joining the cycle. Even the oxygen you breathe has its role here, acting as the final electron acceptor that allows the whole process to keep turning. Without oxygen, the chain backs up, NADH can't unload its electrons, and the cycle stalls. And yet, despite its complexity, there's a calm efficiency to it. The TCA cycle doesn't race or stutter, it hums. Each enzyme knows its place, each intermediate appears and disappears with perfect timing. It's not a frantic scramble for energy, but a graceful rotation, a molecular Ferris wheel turning endlessly in the background of life. Every breath you take fuels this wheel. Every morsel of food you eat is eventually funneled into it. And with every turn, carbon dioxide drifts away into the air, while energy, stored neatly in ATP, flows into the quiet machinery of your cells. The TCA cycle may be invisible, but its rhythm never stops. It is the steady pulse of metabolism, the circular path that turns food into life. Deep inside each of your cells lies a world of quiet machinery, and at the heart of that world, hidden within the folds of the mitochondria, hums one of biology's most extraordinary inventions, the electron transport chain. It's a process so elegant and efficient that it powers almost everything you've ever done. Every heartbeat, every thought, every blink of an eye. This is where energy truly earns its wings, transformed from invisible electrons into the chemical currency of life. The stage for this intricate performance is the inner mitochondrial membrane. Unlike the outer membrane, which is smooth and permeable, the inner membrane is folded into thousands of ridges called cristae, increasing its surface area dramatically. These folds create the workspace where the electron transport chain, or ETC, carries out its silent labour. The process begins with 2 of the cell's best energy couriers, NADH and FADH2, both created earlier in glycolysis and the TCA cycle. They've been patiently holding on to high-energy electrons, waiting to deliver them to this final step. When they arrive, they hand over their electrons to a sequence of large protein complexes embedded in the membrane, each one passing them along like runners in an invisible relay race. There are 4 major complexes, numbered 1 through 4, and 2 smaller but vital helpers. coenzyme Q and cytochrome C. Together they form a chain of redox reactions, a cascade of electron transfers that releases small packets of energy at each step. This is not energy in the sense of heat or motion, but the subtle shifting of electrical potential, harnessed with exquisite precision. Complex I, known as NADH dehydrogenase, accepts electrons from NADH, oxidizing it back into NAD+, and sending the electrons deeper into the chain. As it does, the energy released is used to pump protons, hydrogen ions, from the mitochondrial matrix into the narrow space between the inner and outer membranes. This begins to build an electrochemical gradient, a sort of molecular pressure difference that will soon become immensely important. Next, complex II, or succinate dehydrogenase, joins the process, accepting electrons from FADH2, which provides slightly less energy than NADH. Complex II doesn't pump protons itself, but it passes the electrons to a small, mobile molecule called coenzyme Q, or ubiquinone. Coenzyme Q acts as a courier, ferrying electrons between complexes, gliding effortlessly through the oily interior of the membrane. The electrons then reach complex III, or cytochrome BC1 complex, which transfers them to another carrier, cytochrome C. Cytochrome C is a small water-soluble protein that shuttles electrons one at a time to complex 4, also known as cytochrome C oxidase. Each of these handoffs is a tiny drop in potential energy, but across thousands of such transfers every second, the total yield is immense. Complex 4 is where the journey ends, and where oxygen finally makes its entrance. Oxygen waits patiently at the finish line, accepting the electrons and combining with hydrogen ions to form water. It's a modest role, but without it, the entire system grinds to a halt. Oxygen is the ultimate electron acceptor, the sink into which all the energy of food finally drains. This is why you breathe, not to take in oxygen for its own sake, but to keep this quiet electron traffic flowing. If oxygen runs low, electrons can't exit the chain. They pile up, NADH and FADH2 remain reduced, and earlier reactions, glycolysis and the TCA cycle, slow down as a result. The cell shifts to emergency measures like fermentation, producing ATP less efficiently until oxygen returns. In the absence of oxygen, it's as if the entire city's power grid is dimmed. Lights flicker, work slows, but the system hangs on, waiting for the current to resume. While electrons travel through the chain, something even more remarkable is happening alongside them. Each complex that pumps protons across the inner membrane is quietly charging the system, building up what's called a proton motive force, a combination of a chemical gradient difference in proton concentration and an electrical gradient difference in charge. The matrix becomes slightly negative and less acidic, while the intermembrane space becomes more positive and proton-rich. It's as if the mitochondrion is filling a tiny reservoir of potential energy, patiently storing it across a membrane only a few nanometers thick. And then comes the payoff. The protons that have been pumped out now want to come back in. Nature abhors imbalance. But the membrane allows them only one way home, through a spectacular molecular machine called ATP synthase. This enzyme is part turbine, part generator, and all elegance. As protons flow through it, they cause part of the structure to rotate, literally spin as within the membrane. This mechanical motion drives the chemical reaction that joins ADP and inorganic phosphate to form ATP. It's almost poetic. Chemistry becomes motion, and motion becomes chemistry again. A physical rotation produces a chemical bond, linking the mechanical and the molecular in perfect harmony. This process, known as chemiosmosis, was once a controversial idea proposed by the scientist Peter Mitchell, who was initially met with skepticism. Today it stands as one of the most beautiful principles in biology, that cells use tiny proton gradients to power their entire existence. Each turn of ATP synthase produces roughly 3 molecules of ATP. The enzyme is astonishingly efficient, converting over 90% of the proton motive force into usable energy. Under an electron microscope, it looks like a small mushroom-shaped knob studded on the mitochondrial membrane, and though it's far smaller than a speck of dust, it spins at several hundred revolutions per second. You could call it the world's tiniest water wheel spinning in the dark, driven not by a river, but by a stream of protons, each flow creating a whisper of motion that keeps the lights of life on. What's truly mind-bending is how quiet all this is. Millions of these rotary motors are turning right now in your body, producing the ATP that fuels every heartbeat and every neuron firing in your brain. And yet, you don't feel it. There's no heat, no vibration, just the soft hum of molecular industry as seamless as breathing. The efficiency of this system is staggering. From a single molecule of glucose, cellular respiration can yield up to 32 molecules of ATP, most of them generated here in this final stage. The earlier pathways prepared the ingredients, but this is where the cell finally cashes in. The electron transport chain is also carefully protected. Too much electron leakage can lead to the formation of reactive oxygen species, highly reactive molecules that can damage proteins and DNA. To counter this, mitochondria carry an array of antioxidants and repair systems that keep the balance in check. Even here, at the molecular level, life is a game of precision. A little energy too quickly released, and the system risks burning itself out. When everything's in balance, the result is a masterpiece of coordination. Electrons flow like a gentle current. Protons drift through ATP synthase, and ATP appears in steady supply. Energy, order, and life sustained through movement smaller than imagination. You could think of the mitochondrion as a microscopic power plant, but that comparison doesn't quite do it justice. Unlike human-made engines, this one never sputters, never overheats, and never sleeps. It's been running in almost every living cell on Earth for billions of years, spinning its invisible turbines through every second of your life. Every breath you take refills the system with oxygen. Every exhale carries away carbon dioxide, the echo of reactions that happened deep inside you moments before. It's an ancient rhythm. Electrons fall, protons flow, ATP forms, and life continues, one silent revolution at a time. If proteins are the workers of life and carbohydrates the quick fuel, then lipids are the quiet architects, shaping boundaries, storing energy, and sending messages across the invisible landscapes of cells. They are the molecules that define what is inside and what is out, yet they never build walls. Instead, they make membranes, flexible, fluid barriers that hold everything together without ever holding anything still. At first glance, lipids seem simple. They're made mostly of carbon and hydrogen, with just enough oxygen to add a touch of character. Yet from this simplicity arises enormous variety. The most familiar are fatty acids, long chains of carbon atoms capped with a carboxyl group at one end and a tail of hydrogens at the other. They can be saturated, with no double bonds and therefore straight and tightly packed, or unsaturated, where one or more double bonds create gentle kinks that prevent tight packing. This small difference determines whether a fat is solid like butter or liquid like olive oil at room temperature. Link 3 fatty acids to a glycerol backbone and you get a triglyceride, the main form of stored fat in animals. It's compact, stable, and wonderfully energy-rich. 1 gram of fat stores more than twice the energy of a gram of carbohydrate, which makes it nature's favourite long-term fuel. Plants tuck triglycerides into seeds, animals store them in adipose tissue, and migratory birds rely on them to cross oceans without refueling. For humans, that soft layer under the skin or around organs is not just insulation, it's a quiet reservoir of energy, ready to be mobilized when food is scarce. But lipids don't stop at storage, they also build structure. The stars of this show are phospholipids, the molecules that make up cell membranes. A phospholipid looks like a tadpole with a dual personality, a hydrophilic head that loves water and hydrophobic tails that avoid it. When placed in water, they spontaneously arrange themselves into bilayers, with the heads facing outward toward the water and the tails tucked safely inside. The result is the phospholipid bilayer, the fundamental structure of all cellular membranes. It's a self-assembling miracle, no instructions required. Yet despite appearing sturdy, membranes are not rigid. They are fluid, shifting and moving constantly like the surface of a pond. Proteins drift within them, lipids slide past one another, and small regions thicken or thin depending on what's happening inside the cell. This fluidity is crucial. Without it, cells couldn't grow, divide, or communicate. The degree of fluidity depends on several factors. The saturation of fatty acids plays a key role. Membranes with more unsaturated tails are looser and more flexible, while those with saturated tails are stiffer and less permeable. Temperature matters too. At colder temperatures, membranes can become too rigid, so organisms in chilly environments often pack their membranes with unsaturated fats to keep them fluid. Conversely, in heat, too much fluidity can be dangerous, so cells increase saturation to keep their structure stable. Then there's cholesterol, the unlikely hero of membrane physics. It's a sterol, a type of lipid with a rigid ring structure that fits snugly among phospholipids. Cholesterol acts like a molecular shock absorber, preventing membranes from freezing solid in the cold and from becoming too floppy in the heat. In just the right amount, it makes membranes both strong and supple. Without cholesterol, animal cells would lose their graceful balance, and even the simplest movements of life would become sluggish or erratic. Within this fluid landscape, membranes are far from uniform. They contain microdomains known as lipid rafts, tiny patches enriched with cholesterol, sphingolipids, and specific proteins. These rafts act like floating platforms, organizing signaling molecules and serving as staging areas for communication between the inside and outside of the cell. Imagine them as small meeting points on the cell's surface, where molecules gather to exchange information before dispersing again. The existence of these rafts adds another layer of complexity to membrane dynamics, a reminder that even at the molecular level, organization and movement coexist in perfect harmony. Membranes are also dynamic in their shape. They curve, fold, and fuse, forming vesicles that transport materials from one part of the cell to another. Specialized proteins can bend membranes into bubbles or pinch them off entirely, creating the cargo packets that carry neurotransmitters, hormones, and other molecules. The flexibility of lipid bilayers allows cells to reshape themselves constantly, dividing, engulfing, communicating, and adapting to the world around them. And while membranes form the visible frontier of life, lipids continue to play vital roles behind the scenes. Some act as signals, carrying messages between cells or within them. Lipid-derived messengers such as prostaglandins and leukotrienes help regulate inflammation, immunity, and even pain perception. Others, like diacylglycerol, participate directly in signal transduction, helping relay messages from the cell surface to its interior machinery. These lipid signals are short-lived but powerful, proving that even the quietest molecules can start a biochemical conversation that changes the fate of a cell. Lipids also play a starring role in energy metabolism. When the body needs to draw on stored fat, beta-oxidation takes over, a process that occurs inside the mitochondria. Fatty acids are broken down step by step, removing 2 carbon units at a time to generate acetyl-CoA, which then enters the TCA cycle for further oxidation. Each turn of this process yields high-energy electrons, captured by NADH and FADH2, which feed directly into the electron transport chain to produce ATP. Compared to carbohydrates, fats release more than twice as much energy per gram, which is why marathon runners and migrating animals depend so heavily on them once short-term glycogen stores are depleted. This remarkable efficiency, however, comes at a cost. Fat metabolism requires more oxygen than carbohydrate metabolism, meaning it's slower under high-intensity conditions. During a sprint, for instance, muscles rely on glycolysis for quick bursts of ATP, because oxygen delivery can't keep up with demand. But in endurance activity, or quiet rest, beta oxidation takes the lead, steadily supplying energy over the long haul. It's the difference between a match flare and a slow enduring flame. Lipids are also involved in temperature regulation, buoyancy, and even protection. Whales and seals rely on thick layers of blubber for insulation in icy oceans, while certain plants adjust their membrane lipids seasonally to cope with frost or heat. In humans, fat cushions organs, protects nerves, and stores fat-soluble vitamins like A, D, E, and K, a perfect example of how lipids quietly support life on multiple levels. Despite their bad reputation in diet debates, lipids are indispensable. Without them, cells would fall apart, energy would vanish, and communication would cease. Even cholesterol, often vilified, is essential for producing hormones such as estrogen and testosterone, and for stabilizing membranes in every cell. The key is not elimination, but balance, the same principle that governs every aspect of metabolism. If you could zoom into a cell membrane, it would look like a shimmering mosaic, lipids drifting in a gentle dance, proteins bobbing among them, and small patches of organization forming and dissolving. There's no solid wall here, no static barrier. Instead, the cell's boundary is alive, constantly changing, responding, and adapting. The beauty of lipids lies in this paradox. They create boundaries that protect life, yet those boundaries are flexible enough to allow life to flow through them. They hold everything together while letting everything move. It's a quiet, elegant balance, one that keeps the cell safe, responsive, and endlessly alive. Every living cell exists on a border, a thin edge between what is inside and what is out. That edge, the cell membrane, is not a wall, but a living frontier. Molecules move across it constantly, slipping in and out, balancing, exchanging, and communicating. Life depends on this movement, on the careful management of difference. Gradients, those subtle imbalances of concentration, charge, or pressure, are not imperfections in biology. They are its driving force. Life, as it turns out, loves an edge. At the simplest level, movement across the membrane can happen in two ways: passive or active transport. Passive transport requires no energy from the cell. Molecules move naturally from areas where they are more concentrated to where they are less, a process called diffusion. It's as if molecules are simply evening out the crowd, drifting toward open space until balance is achieved. Oxygen, carbon dioxide, and small lipophilic molecules use this easy route, slipping through the lipid bilayer without assistance. But most substances in the cell are not so lucky. Ions, sugars, and amino acids are too polar or too large to cross unaided. To help them, cells employ proteins that act as molecular doorways. This is facilitated diffusion, movement that still follows the gradient but with a little assistance. There are two main types of helpers: channels and carriers. Channels form tiny pores that open and close, allowing specific ions or water molecules to flow through. They're the fast lanes of the membrane, letting thousands of particles pass each second. Some are always open, while others, known as gated channels, respond to signals like voltage, pressure or chemical messengers. Carriers, on the other hand, are more selective and deliberate. They bind their passenger, change shape to carry it across, and then release it on the other side. It's slower but more controlled, like a revolving door rather than an open gate. This quiet choreography of movement keeps the cell in balance, maintaining the right levels of nutrients, ions, and water, but sometimes the cell needs to move substances against the gradient, from low concentration to high, which requires effort. That's where active transport comes in, powered by energy in the form of ATP or by the movement of other ions. In primary active transport, the energy comes directly from ATP. The best-known example is the sodium-potassium pump, a protein that works tirelessly in almost every cell. For every 3 sodium ions it pushes out, it brings 2 potassium ions in. This constant pumping maintains steep gradients across the membrane, with high sodium outside and high potassium inside. These gradients are vital for nerve impulses, muscle contractions, and even the basic electrical potential of the cell. The pump itself is a masterpiece of molecular engineering. It changes shape as it binds and releases ions, using the energy from ATP hydrolysis to drive the motion. Then there is secondary active transport, which doesn't use ATP directly, but takes advantage of the gradients established by primary pumps. Think of it as using one stored form of energy to power another task, like water flowing downhill turning a wheel that lifts more water uphill. A perfect example of this is the sodium-glucose cotransporter, found in your intestines and kidneys. As sodium ions flow back into the cell down their concentration gradient, the transporter harnesses that energy to pull glucose molecules in with them, against their own gradient. The sodium is happy to move down its slope, and glucose gets a free ride uphill, both passing through in the same direction, a process known as symport. The beauty of this system is its efficiency. No ATP is spent directly, yet energy is still captured from the careful management of gradients. Gradients, in fact, are one of life's most versatile tools. They store energy and carry information. When an electrical charge difference builds up across a membrane, it forms what biologists call a membrane potential, a tiny voltage that cells use like a battery. Nerve cells, for example, exploit these voltages to send signals. When a neuron fires, ion channels open in rapid sequence, allowing sodium to rush in and potassium to flow out. The resulting wave of charge travels along the cell's membrane like a whisper of electricity, passing information faster than any chemical messenger could alone. Elsewhere, gradients serve as guides. Bacteria can sense chemical gradients in their surroundings and swim toward food or away from toxins. Inside multicellular organisms, gradients of ions, signaling molecules, or pH help cells determine where they are and what they should do. Even embryonic development, the process of shaping an entire organism from a single cell, relies on subtle gradients that tell each cell which genes to activate and what structure to form. The most familiar and perhaps most misunderstood form of gradient-driven movement is osmosis, the flow of water. Water, the universal solvent, always tries to balance concentrations across a barrier. If solutes, salts, sugars, or proteins, are more concentrated on one side, water moves toward that side to dilute them. It's nature's way of equalizing differences. Many people imagine osmosis as water seeking salt, but that's not quite right. It's really about probabilities. Water molecules are constantly moving, and more of them happen to leave the side with fewer solutes, since there are fewer obstacles in the way. The result is a net flow toward the more crowded side until equilibrium is reached. However, water doesn't slip through the lipid bilayer very well on its own, so cells rely on specialized protein channels called aquaporins. These are narrow pores lined with hydrophilic groups that let water pass single file, one molecule at a time, while blocking ions and other substances. Aquaporins are the cell's sluice gates, regulating the gentle flow of water that keeps tissues hydrated and cells properly swollen. In your kidneys, for example, aquaporins are constantly opening and closing to fine-tune water balance, helping you conserve or release water depending on your body's needs. The subtlety of all this motion is astonishing. Ions, sugars, and water drift through membranes at speeds and quantities that would seem chaotic, yet the result is perfect order. The cell maintains its shape, its electrical balance, and its concentration gradients, all through this interplay of passive and active transport. It's like a self-regulating city of doors and pumps, each one responding to signals in real time. This balancing act also shows how life thrives on differences. A cell without gradients is a cell without energy or direction. When everything inside and outside becomes equal, the flow stops, and with it, life itself. Maintaining that edge, that controlled imbalance, is what keeps every system in motion. It's easy to overlook the elegance of something as mundane as diffusion or osmosis. But these are the quiet forces behind everything, from nerve impulses to nutrient absorption. They are the invisible tides that make life dynamic. Every breath you take, every drop of sweat, every beat of the heart depends on these gradients, tiny, persistent slopes of energy and concentration that define what it means to be alive. In the end, cells don't build high walls or seal themselves off. They build borders that breathe. They let the world in and out carefully, constantly, keeping themselves just slightly out of balance, just enough to stay alive. Because life in all its complexity depends not on stillness, but on the graceful flow along its edges. Every living cell is a workshop, and its most versatile tools are proteins, long chains of amino acids that twist, fold, and assemble into forms so varied and precise that nearly every task of life depends on them. Enzymes, hormones, receptors, transporters, antibodies, all of them are proteins, each built from the same basic alphabet of 20 amino acids. The variety comes not from the number of letters, but from the infinite ways they can be arranged and folded. Each amino acid has the same simple backbone, a central carbon attached to an amino group, a carboxyl group, a hydrogen, and a distinctive side chain, the R group. That side chain is what gives each amino acid its personality, from the water-loving serenity of serine to the fiery reactivity of cysteine. Some are polar, some non-polar, some are acidic, some basic. Together, they create a delicate chemistry of attraction and repulsion, driving proteins to fold into the shapes that define their function. It helps to think of these side chains as tiny characters in a drama. The hydrophobic ones, valine, leucine, isoleucine, prefer to hide together in the interior, away from water. The charged ones, lysine and glutamate, like to stand near the surface, interacting with the watery environment or forming salt bridges with each other. Aromatic residues such as phenylalanine or tryptophan add stability through stacking interactions, while small, flexible amino acids like glycine bring movement and grace. Each one plays a role, and the pattern in which they appear along the chain determines how the story unfolds. That sequence, the exact order of amino acids in a chain, is known as the primary structure of a protein. It might look like a meaningless string of letters to the untrained eye, but it carries all the information the molecule needs to fold itself into a specific functional shape. The cell doesn't carve proteins into form. It lets them assemble themselves, guided by the laws of chemistry and the quiet push of thermodynamics. From that primary structure arise higher levels of organization. As the chain begins to twist and coil, it forms secondary structures, patterns such as alpha helices and beta sheets. Alpha helices are tight, spring-like coils stabilized by hydrogen bonds, while beta sheets are flatter, formed when segments of the chain lie side by side and link together through the same kind of bonding. You can imagine alpha helices as ribbons curling gracefully, and beta sheets as the folds of a pleated fabric. These structures provide strength and flexibility, turning simple sequences into recognizable forms. As the chain continues to fold, those helices and sheets pack together into a tertiary structure, the full, three-dimensional shape of a single protein molecule. This stage is like watching origami come to life. The folds and tucks create pockets, channels, and surfaces that allow the protein to interact with other molecules. Here, function finally emerges. A small cavity might become an enzyme's active site. A surface groove might recognize DNA. A flexible loop might act as a hinge for motion. Some proteins stop there, perfectly functional as single units. Others join together, forming complexes known as quaternary structures. Hemoglobin, for example, is made of 4 subunits that work in harmony, each one able to bind oxygen. These assemblies bring new capabilities: cooperation, regulation, and resilience, much like a team achieving what no individual could. The folding process itself is a masterpiece of self-organization. Proteins don't wander aimlessly into shape. They follow a folding funnel, a conceptual landscape where countless possible forms gradually narrow toward the most stable configuration. Along the way, weak interactions guide the molecule: hydrogen bonds, van der Waals forces, hydrophobic clustering, each one gently nudging the chain toward its final state. Yet even this graceful process sometimes needs help. Inside cells, specialized helper proteins called chaperones ensure that folding happens correctly. They don't dictate the final structure, but they provide a safe space where newly made proteins can fold without tangling or sticking to the wrong partners. Imagine a quiet backstage area where delicate origami can be folded away from gusts of wind and sticky fingers. When folding goes wrong, the consequences can be serious. Misfolded proteins may clump together, forming insoluble aggregates that the cell struggles to clear. Some of these aggregates, such as amyloid fibrils, are associated with diseases like Alzheimer's and Parkinson's. These fibrils are stubbornly stable, forming long sheet-like structures that resist breakdown. It's one of nature's tragic ironies that the same forces that create strong functional proteins can also create damaging, unbreakable ones when the balance slips. Still, for every misfolded mistake, there are countless examples of perfection. Proteins folding with astonishing speed and precision, often in less than a second. This reliability is what allows life to function at all. It's also what allows evolution to explore endless variations. A single change in one amino acid can subtly tweak a protein's shape, giving rise to new functions or adaptations. One of the most elegant examples of how protein structure and function intertwine is hemoglobin, the oxygen carrier in your blood. Each hemoglobin molecule is a tetramer, 4 subunits, each holding a heme group with an iron atom at its center. This iron atom binds oxygen, but not too tightly. If hemoglobin clung to oxygen with desperation, it would never let go. If it held it too loosely, it couldn't pick it up. The secret lies in its allosteric behavior, its ability to change shape in response to binding. When one subunit binds oxygen, it slightly shifts the position of its neighbors, making it easier for them to bind oxygen as well. This phenomenon, called cooperative binding, means that hemoglobin loads oxygen efficiently in the lungs, where oxygen is abundant, and releases it easily in tissues where oxygen is scarce. It's a molecular version of teamwork, each subunit encouraging the next, adjusting and responding as conditions change. This same principle of allostery, structural change leading to functional change, appears throughout biochemistry. Enzymes switch between active and inactive forms depending on what binds to them. Receptors alter their shape when a hormone docks. Ion channels open or close as voltage shifts across a membrane. Proteins are not rigid machines. They're flexible instruments, playing the tune of chemistry through motion and adaptation. The elegance of this system lies in its balance between structure and movement. A protein's stability depends on its shape, but its usefulness depends on its ability to change that shape when needed. It's the difference between a sculpture and a living muscle. One is fixed, the other alive. Proteins live at that intersection, stable enough to persist, flexible enough to respond. When you step back, it's remarkable how much of life's complexity arises from a simple alphabet of 20 amino acids. From silk to skin, enzymes to antibodies, it's all variations on the same theme, chains of atoms folding into meaning. The cell is filled with these molecules in motion, bending, flexing, binding and releasing in endless cycles. Proteins are not just the machinery of life, they are its poetry, built from repetition, rhythm and form, and all of it begins with the chemistry of a few humble side chains, linking together, folding gracefully, and finding purpose in their final shape. If you could peer into the heart of every cell, you'd find something both simple and profound, a molecule that carries all the instructions for life, written in a language only chemistry can read. That molecule is DNA, deoxyribonucleic acid, and despite its fame, it's astonishingly modest in appearance. It isn't glowing, it isn't alive, and yet it holds the design for everything that is. At first glance, DNA looks like a long twisted ladder, a double helix. Each side of the ladder is made of alternating sugar and phosphate molecules, forming the sturdy backbone. The rungs in between are pairs of nitrogenous bases, which come in 4 types: adenine, thymine, cytosine, and guanine. The beauty of DNA lies in its rule of pairing. Adenine always pairs with thymine, and cytosine always pairs with guanine. This perfect complementarity is held together by hydrogen bonds, 2 between A and T, 3 between C and G. Its chemistry is poetry, precise, balanced, and endlessly repeatable. The 2 strands of the double helix run in opposite directions, a feature known as anti-parallel orientation. One strand runs from the sugar's 5th carbon to its 3rd. The other runs the reverse way, from 3 to 5. This arrangement seems like a small detail, but it's what allows the molecule to replicate and repair itself efficiently. You could think of it like 2 lanes on a winding road, side by side heading in opposite directions, perfectly aligned for traffic flow. DNA structure isn't just beautiful, it's practical. The sequence of bases, A, T, C and G, forms the instructions for building proteins, the machinery of life. But the molecule itself is also incredibly stable. The phosphate backbone shields the bases inside, and the hydrogen bonds give just enough flexibility for the strands to separate when needed. Stability with accessibility, it's a delicate balance, mastered over billions of years. Now, if DNA were left as a long, loose molecule, it would be chaos. In a single human cell, the DNA, if stretched out, would measure about 2 meters long, yet it fits neatly inside a nucleus barely a few micrometers wide. The trick lies in chromatin, the way DNA is packaged. The molecule wraps around clusters of proteins called histones, forming structures that resemble beads on a string. Each bead is a nucleosome, and together they coil and loop into higher levels of organization, compacting the DNA thousands of times over. This packaging is not just for storage, it's for control. Loosely packed regions, called euchromatin, are active. Genes here can be read and expressed. Densely packed regions, heterochromatin, are more silent, keeping certain genes off or protecting structural regions like telomeres. The cell constantly adjusts these levels of packaging depending on what's needed, tightening some regions, relaxing others. It's like a well-organized library where some books are on the desk, open and ready to read, while others rest quietly on the shelves. When it's time to divide, the cell must make a complete copy of its DNA, an astonishing feat of precision known as replication. The process begins at specific sites called origins of replication, where enzymes pry open the double helix, forming a replication fork, a Y-shaped structure where the strands separate and new ones are built. Here, an enzyme called DNA helicase acts like a zipper pull, unwinding the strands and breaking the hydrogen bonds between bases. But unwinding DNA creates tension ahead of the fork, so another enzyme, topoisomerase, steps in to relieve the strain, preventing tangles and supercoils. Meanwhile, single-strand binding proteins coat the exposed strands, keeping them from snapping back together. Once the template strands are open, the real work begins. DNA polymerase, the star enzyme of replication, adds new nucleotides to build complementary strands. But it can only add to an existing chain. It can't start one from scratch. That's where a short RNA piece called a primer comes in, laid down by Primus to give polymerase a starting point. Replication happens in two distinct ways on the separated strands, thanks to their antiparallel orientation. One strand, called the leading strand, is synthesized continuously, following the helicase as it unwinds the DNA. The other, the lagging strand, runs in the opposite direction, so it's built in short fragments known as Okazaki fragments. Each fragment starts with its own RNA primer and is later joined together by DNA ligacy to form a seamless strand. The coordination of these enzymes is astonishing, like a well-rehearsed dance with helicase unwinding, polymerase building, and ligase stitching everything together. The entire process proceeds at a blistering speed, copying billions of bases in just a few hours, all while maintaining extraordinary accuracy. That accuracy comes from the meticulous proofreading abilities of DNA polymerase. As it adds each nucleotide, it double-checks that the base matches its partner on the template strand. If it makes a mistake, inserting the wrong base, it pauses, removes the error with its built-in editing function, and replaces it correctly. Thanks to this proofreading, DNA replication makes fewer than one error in 10 billion bases. That's the kind of reliability that keeps life stable across generations. Still, perfection is neither possible nor desirable. Occasionally mistakes slip through. A misplaced base here, a small deletion there. These mutations may sound alarming, but they are the seeds of evolution. Most are harmless, some are harmful, but a very few, introduce changes that can be useful. A single altered base might make a protein fold differently, function better, or adapt to new conditions. Without this tiny trickle of change, life would stagnate, unable to evolve or diversify. Cells, of course, have backup systems to keep mutations in check. Enzymes patrol the DNA, scanning for irregularities, nicks, or mismatches. If they find damage, from radiation, chemicals, or simple copying errors, they excise the faulty region and fill it in using the undamaged strand as a template. These repair systems are the quiet custodians of genetic integrity, ensuring that the library of life stays legible even after billions of readings. When DNA replication goes right, it's almost magical. Two identical double helices emerge from one, each ready to be passed on to daughter cells. The entire process is semi-conservative, meaning each new DNA molecule keeps one old strand and builds one new one. It's both a preservation and a renewal, a perfect metaphor for life itself. Under a microscope, the replication forks sparkle like factories at work. Strands unwind, enzymes shuttle, and newly synthesized DNA spools out in elegant symmetry. It's hard to imagine that something so intricate happens millions of times a day inside you, invisibly and flawlessly. And yet, in the calm hum of this molecular machinery lies something deeply poetic. DNA is both fragile and enduring. Its code can survive for thousands of years, yet it's rewritten in every generation, every cell division, every act of life. Those twisting strands, carrying memories from ancestors, instructions for the present and potential for the future, are a reminder that stability and change coexist at the heart of biology. DNA holds the past, but it also whispers the possibilities of what's yet to come. If DNA is the library of life, then RNA is its librarian, the one who reads the books, copies the important parts, and delivers instructions to where they're needed. It's the molecule that bridges the quiet storage of genetic information and the bustling world of proteins and enzymes. DNA may hold the master blueprint, but RNA is the one that makes sure the plans are followed. There's a beautiful simplicity to it. RNA, or ribonucleic acid, is made of the same kind of nucleotides as DNA, only with a few subtle twists. Instead of deoxyribose, its sugar is ribose, which has one more oxygen atom. And instead of thymine, RNA uses uracil, a base that pairs with adenine just as easily. But that small difference gives RNA a different personality. It's less stable, more flexible, and better suited to being a messenger than a volt. There are several kinds of RNA, each with its own specialty. The most famous is messenger RNA, or mRNA, which carries the instructions for building proteins. It's a working copy of a gene, transcribed directly from DNA and read by the ribosome, the cell's protein-making factory. Then there's transfer RNA, or tRNA, which acts as a molecular courier. Each tRNA carries one specific amino acid and matches it to the right spot on the mRNA sequence, like a translator converting words into actions. Another essential player is ribosomal RNA, or rRNA, the structural and catalytic core of the ribosome itself. Unlike DNA, which mostly delegates, RNA can fold into complex three-dimensional shapes and even perform chemical reactions. In fact, it's the RNA, not the protein part of the ribosome, that actually stitches amino acids together. Life's oldest machines, it seems, are made of RNA. Beyond these main types, cells also rely on a constellation of smaller RNAs that fine-tune their activity. MicroRNAs and small interfering RNAs act as regulators, binding to messenger RNAs and preventing them from being translated, or tagging them for destruction. Others, like small nuclear RNAs, help with RNA processing inside the nucleus. Altogether, they form a web of control that keeps gene expression balanced, ensuring the right proteins are made in the right amounts at the right times. The story begins with transcription, the process of copying DNA into RNA. It starts when an enzyme called RNA polymerase attaches to a specific region on the DNA known as a promoter. The promoter acts like a signpost that says "start here." But polymerase doesn't find this sign on its own. It's guided by transcription factors, proteins that recognize particular sequences and recruit the polymerase to the right spot. Once everything is in place, the enzyme pries open the DNA double helix and begins reading one of the strands, adding complementary RNA nucleotides as it goes. The process is surprisingly smooth. RNA polymerase glides along the DNA, unwinding it ahead and sealing it behind, building an RNA strand one base at a time. Where the DNA has a guanine, RNA adds a cytosine. Where the DNA has an adenine, RNA adds uracil. It's like reading from one language and writing in another. The message stays the same, just in a new form. When transcription ends, the RNA polymerase releases the newly made transcript. But for eukaryotes, the story doesn't stop there. The raw RNA, called a prime-RNA, still needs editing. It contains stretches called introns, non-coding regions that must be removed, and exons, which contain the real instructions. A complex molecular machine called the spliceosome cuts out the introns and joins the exons together, producing a clean, readable mRNA. This splicing step is more than just housekeeping. It allows for creativity. Many genes can be spliced in different ways, producing slightly different proteins from the same original transcript, a trick known as alternative splicing. It's one of the reasons a single genome can produce such astonishing diversity. Once the mRNA is complete, it's given a protective cap at one end and a tail of adenine bases at the other, both of which help it survive the journey out of the nucleus. It then travels into the cytoplasm where the ribosomes wait. Here begins translation, the transformation of the RNA message into a working protein. The ribosome itself is a marvel of molecular architecture, a massive complex made of both proteins and RNA, divided into two subunits that fit together like the halves of a clamshell. As the mRNA threads through, the ribosome reads it in sets of 3 nucleotides called codons. Each codon corresponds to one amino acid. The start of the message is marked by a specific codon, AUG, which codes for the amino acid methionine. It's the universal begin signal for protein synthesis. As each codon appears, a matching tRNA arrives, carrying its corresponding amino acid. Each tRNA has a 3-base region called an anticodon, which pairs with the codon on the mRNA like 2 puzzle pieces. When the match is made, the ribosome catalyzes the formation of a peptide bond between the growing chain and the new amino acid. Then the ribosome shifts 3 bases forward, 1 codon, and the next tRNA arrives. Step by step, the chain grows, folding as it emerges from the ribosome's tunnel, already hinting at its final structure. It's an intricate dance. tRNAs moving in and out, the ribosome ratcheting along, enzymes keeping the pace. The energy for all this motion comes from GTP, a cousin of ATP. Every step, from loading tRNAs to moving the ribosome, costs energy. Making a single protein is an expensive task, and yet the cell performs it millions of times a minute, continuously maintaining and renewing its molecular population. When the ribosome reaches a stop codon, one of 3 sequences that signal the end of the message, there's no matching tRNA to deliver an amino acid. Instead, a release factor binds, prompting the ribosome to let go of the completed protein. The chain folds fully. The ribosome separates into its subunits, and the mRNA may go on to be read again or eventually degraded when its work is done. Despite its complexity, translation is remarkably accurate. The genetic code is read with near-perfect fidelity, thanks to proofreading at multiple stages, by tRNA synthetases, which charge each tRNA with the correct amino acid, and by the ribosome itself, which double-checks base pairing before forming bonds. Mistakes do happen, but rarely, about 1 error per 10,000 amino acids, and when they do, the cell often degrades the faulty protein before it can cause trouble. The entire system is so refined that even slight interference can be devastating, which is why many antibiotics work by targeting bacterial ribosomes. Drugs like tetracycline and streptomycin bind to bacterial ribosomal sites, blocking translation or introducing errors. Human ribosomes, though similar, are just different enough that they usually escape harm. It's a reminder that the machinery of life, though ancient and universal, has evolved subtle variations across species, enough for medicine to exploit without breaking the delicate balance of our own cells. There's something deeply poetic about the whole process. In transcription, DNA's silent code is given a voice through RNA. In translation, that voice becomes action, enzymes, hormones, structures, and signals. Every heartbeat, every thought, every movement of a muscle is, at its root, a story being read and performed by RNA and ribosomes. And while DNA is the steadfast archive of life's memory, RNA is its restless interpreter, short-lived but endlessly active, a molecule in motion, turning static information into the music of metabolism. Every cell, at every moment, hums with this rhythm, a chorus of transcription and translation, tirelessly singing life into existence. Cells are constantly talking, not in words or sounds, but in molecules, ripples and flashes of chemistry. Every second, trillions of signals are sent and received within your body, hormones drifting through the bloodstream, neurotransmitters hopping across tiny gaps between neurons, and growth factors brushing gently against cell membranes. Together they form a grand conversation that keeps tissues coordinated, organs synchronized, and life in motion. At the surface of every cell are thousands of receptors, the molecular equivalents of doorbells. Each one is tuned to recognize a specific signal, a ligand, that fits it perfectly, like a key sliding into a lock. When that ligand binds, it doesn't enter the cell directly. Instead, it rings the bell, triggering a chain reaction inside. These receptors are the first translators between the outside world and the cell's inner workings. Two of the most important families of receptors are the G-protein coupled receptors, GPCRS, and the receptor tyrosine kinases, RTKS. GPCRS are everywhere, in your senses, your brain, your heart, and they respond to an astonishing variety of signals from adrenaline to light itself. Each GPCR threads back and forth across the membrane 7 times, like a ribbon woven through a wall. When a ligand binds on the outside, it changes shape, activating a G protein on the inside of the membrane. This G protein, named for its use of guanine nucleotides, acts like a molecular switch, flipping on when it binds GTP and off when it hydrolyzes it to GDP. Once switched on, the G protein splits into 2 parts that go off to trigger different second messengers, small, fast-moving molecules that spread the signal through the cell. These messengers don't act alone, they amplify. A single receptor binding event can lead to the production of thousands of messenger molecules, turning a tiny external cue into a large, coordinated internal response. One of the best-known second messengers is cyclic AMP, or CAMP. It's made by an enzyme called adenylyl cyclase, which is activated by certain G proteins. When CAMP levels rise, they activate a protein called protein kinase A, or pKa, which in turn phosphorylates a host of other proteins, turning enzymes on or off, opening ion channels, and even altering gene expression. The entire cascade can happen in seconds, allowing the cell to respond almost instantly to changing conditions. Another major signaling route involves calcium ions, or Ca2+, one of the most ancient messengers in biology. Normally, calcium levels in the cytoplasm are kept extremely low. The cell stores it away in the endoplasmic reticulum, like a powerful tool locked in a cabinet. But when a signal arrives, calcium channels open and ions flood out in a brief shimmering pulse. This sudden rise acts as a cue for numerous processes: muscle contraction, neurotransmitter release, even the activation of enzymes. When the job is done, pumps quickly restore order, tucking calcium back into its compartments. Then there are the twin messengers IP3, inositol triphosphate, and DAG, diacylglycerol, born from the same reaction. When certain GPCRS are activated, they stimulate an enzyme called phospholipase C, which slices a membrane lipid into IP3 and DAG. IP3 travels into the cytoplasm and opens calcium channels in the endoplasmic reticulum, while DAG stays in the membrane and activates protein kinase C, PKC. The result is a coordinated surge of activity, calcium and PKC working together to reshape the cell's behavior. In many pathways, these second messengers feed into larger webs of regulation called kinase cascades. A kinase is an enzyme that adds a phosphate group to another protein, altering its activity. In a cascade, one kinase activates another, which activates another, and so on, an elegant chain reaction that multiplies the original signal. These cascades don't just amplify, they integrate. Multiple signaling pathways can converge on the same kinases, allowing the cell to combine information from different sources before deciding what to do. For instance, growth factor signals through receptor tyrosine kinases often lead to the activation of the MAP kinase pathway, a well-studied cascade involved in cell division, differentiation, and survival. When a ligand binds to an RTK, the receptor pairs up with another identical molecule and phosphorylates itself, a process known as autophosphorylation. This creates docking sites for a series of proteins that pass the signal inward step by step until it reaches the nucleus. There, specific transcription factors are activated and the cell's gene expression changes, a long-term adjustment in response to a fleeting message. While these systems may sound like perpetual motion machines, they are anything but. Cells are careful about when to start a signal and just as careful about when to stop it. The same enzymes that add phosphates also have counterparts that remove them, phosphatases, which act as brakes, returning proteins to their resting state. Cyclic AMP is broken down by phosphodiesterases, G proteins deactivate themselves by hydrolyzing GTP, and receptor complexes are often internalized and recycled. It's an elegant system of checks and balances, ensuring that no signal lingers longer than it should. This balance between activation and termination is crucial. Imagine if every adrenaline signal lasted forever. Your heart would never slow down. Or if a growth signal refused to stop, cells would divide endlessly. In fact, the failure to shut off signaling pathways is one of the hallmarks of cancer, where a single broken feedback loop can turn a gentle nudge to grow into a permanent command. The timescales of these signals vary widely. Some, like the opening of an ion channel or the release of calcium, happen in milliseconds. Others, like changes in gene expression, unfold over hours. The difference between a quick pulse of CAMP and a sustained increase can mean the difference between a brief contraction and a full program of cell growth. Cells can even interpret patterns in these signals. Short bursts may trigger one response, while prolonged activation triggers another. It's less like a single on-off switch, and more like a language, rich with tone and rhythm. One of the most remarkable aspects of cell signaling is its sensitivity. A cell can detect and respond to a single molecule of hormone among billions of other molecules. This precision comes not from brute force, but from amplification and feedback. Each step in the cascade magnifies the previous one, ensuring that even the faintest whisper from the outside world is heard loud and clear inside. But cells also know how to quiet themselves once the message has been received. Negative feedback loops are everywhere in signaling. Molecules produced at the end of a pathway often act to shut down the beginning. This prevents overreaction and helps the system reset for the next signal. It's like a party that ends with someone gently turning down the lights and asking everyone to head home. Order restored, but ready for the next celebration. Despite all this complexity, cell signaling feels almost graceful. It's the choreography of molecules. Receptors passing messages to proteins, enzymes lighting up pathways, calcium waves shimmering through the cytoplasm. The signals are transient, but their effects can last a lifetime. Growth, learning, memory, immunity. At its heart, cell signaling is about connection. No cell lives in isolation. Even the simplest bacterium is constantly sensing and responding to its environment. The ability to receive, amplify and regulate signals is what allows life to coordinate, to be more than just a collection of molecules. And all of it begins with a quiet ring at the door, a ligand binding to its receptor and a cascade that unfolds through the cell like a whisper turning into a song. Every leaf on every tree is a small miracle. Quiet, unassuming, and usually taken for granted, it performs one of the most astonishing feats in the natural world, turning light into life. Through photosynthesis, plants, algae, and certain bacteria capture sunlight, split water, and transform carbon dioxide into sugars. It's a process that not only feeds them, but indirectly sustains nearly every living thing on Earth. Deep inside a leaf are tiny green organelles called chloroplasts, descendants of ancient cyanobacteria that were once free-living cells, long ago invited into partnership with their hosts. Each chloroplast is filled with stacks of thin, disc-like membranes called thylakoids, arranged in piles known as grana. These are the solar panels of the cell where the magic begins. Embedded in the thylakoid membranes are clusters of pigments and proteins known as photosystems, the molecular light catchers that start it all. There are two main photosystems, elegantly named photosystem 2 and photosystem 1. The numbering might seem backward, but it reflects the order in which they were discovered, not the order in which they work. In the actual sequence of events, photosystem 2 goes first. When sunlight strikes the chloroplast, photons are absorbed by chlorophyll molecules within photosystem 2. These pigments specialize in capturing red and blue wavelengths, reflecting green. The reason leaves look the way they do. Each photon excites an electron in chlorophyll, boosting it to a higher energy state. The excited electron is then passed to an electron transport chain, a series of carriers that move it along step by step through the thylakoid membrane. But now photosystem 2 has a problem. It's lost an electron and needs another to keep the process going. To replace it, the system does something extraordinary. It splits water. An enzyme complex associated with photosystem 2 pulls electrons from water molecules, releasing hydrogen ions and oxygen as byproducts. This is the origin of nearly all the oxygen in Earth's atmosphere, a byproduct of plants quietly keeping their electron supply topped up. Every breath you take owes itself to this subtle act of molecular generosity. As electrons move along the transport chain, they release energy, which is used to pump protons, hydrogen ions, into the interior space of the thylakoid, known as the lumen. This creates a proton gradient, a build-up of positive charge inside the thylakoid, much like the stored pressure in a dam. Eventually the electrons arrive at photosystem 1, where they're given another boost of energy by another photon of light. The journey these electrons take between the two photosystems is often represented as a Z-scheme, a zigzag line that maps their energy levels over time. First, light from photosystem 2 kicks them up. They lose some energy as they move along the chain, then light from photosystem 1 kicks them up again. It's a molecular relay powered by sunlight, one pulse of energy followed by another, perfectly timed. After their second burst of energy, the electrons are handed off to an enzyme called NADP plus reductase, which transfers them to NADP plus, forming NADPH, one of the cell's high-energy carriers. At the same time, the proton gradient built up in the thylakoid is used to power ATP synthase, the same spinning enzyme found in mitochondria. As protons flow back out of the thylakoid lumen through ATP synthase, the enzyme rotates and joins ADP with inorganic phosphate to make ATP. In short, the light reactions of photosynthesis produce two key molecules, ATP and NADPH, both loaded with energy and reducing power. But the plant isn't done yet. These molecules are only the temporary currency. The real goal is to convert this fleeting energy into stable chemical form, sugar. That happens in the second stage of photosynthesis, the Calvin cycle. The Calvin cycle takes place in the stroma, the fluid-filled area surrounding the thylakoids. Here, carbon dioxide from the air is fixed. meaning incorporated into organic molecules, in a process driven by ATP and NADPH from the light reactions. The key player here is an enzyme called rubisco, short for ribulose 1,5-bisphosphate carboxylase/oxygenase, a name almost as unwieldy as the enzyme itself. Rubisco is both brilliant and flawed. It's the most abundant enzyme on Earth, responsible for capturing vast amounts of carbon from the atmosphere every day. But it's not very fast, and it's not very selective. Sometimes it grabs oxygen instead of carbon dioxide, initiating a wasteful process called photorespiration. Plants have evolved clever ways to cope with this inefficiency. C4 and CAM photosynthetic pathways are examples, but even so, RUBISCO remains a bit of a clumsy hero, essential, ancient, and imperfect. In the Calvin cycle, rubisco attaches carbon dioxide to a 5-carbon sugar called ribulose bisphosphate, producing a short-lived 6-carbon intermediate that immediately splits into 2 3-carbon molecules. Through a series of enzyme-catalyzed steps, these molecules are reshaped and energized using ATP and NADPH, eventually producing glyceraldehyde 3-phosphate G3P. From there, some of the G3P exits the cycle and goes on to become glucose and other sugars, while the rest is recycled to regenerate ribulose bisphosphate, keeping the system running. For every molecule of glucose produced, the Calvin cycle must turn 6 times, fixing 6 molecules of carbon dioxide and consuming 18 molecules of ATP and 12 molecules of NADPH. It's energy intensive, but sunlight provides the fuel in endless supply. The reward is a sugar molecule, a compact, stable form of energy that can be stored, transported, or used to build everything from cellulose to starch. The results of photosynthesis reach far beyond the leaves that perform it. The oxygen released into the air reshaped Earth's history, paving the way for complex aerobic life. Billions of years ago, before photosynthetic organisms evolved, the atmosphere was nearly devoid of oxygen. When cyanobacteria began splitting water and releasing O2, the so-called Great Oxygenation Event transformed the planet, rusting its iron, forming the ozone layer, and eventually making it habitable for animals like us. Every modern ecosystem still depends on this steady flow of photosynthetic energy. The sugars made in plants feed herbivores, herbivores feed predators, decomposers recycle the leftovers, all powered by the energy that started as sunlight on a leaf. Even fossil fuels are ancient sunlight, stored in plant tissues millions of years ago and compressed into oil and coal. It's humbling to realize that every meal, every breath, every flicker of warmth on your skin traces back to the same quiet process in green cells. Photosynthesis also governs the global carbon cycle, balancing the carbon dioxide we exhale or burn with the carbon plants absorb. When forests flourish, they draw down atmospheric carbon. When they vanish, that carbon returns to the air. The rhythm of photosynthesis and respiration is the planet's heartbeat, a vast exchange of gases and energy that links every organism, every ocean, every forest. At the molecular level, it's also small. Photons, electrons, protons, enzymes. But the scale of its impact is cosmic. The light that left the sun 8 minutes ago is, at this moment, landing on a leaf somewhere and being transformed into food, fuel, and oxygen. It's one of the few processes that unites physics, chemistry, and biology in perfect harmony, a single dance connecting the stars to the soil. So next time you walk past a tree or touch a blade of grass, remember, it's not just green pigment you're seeing, it's sunlight, captured and stored in molecular bonds, a slow, patient alchemy that turns starlight into sugar, energy into existence, and light into life. Life is a balancing act. Every moment your cells are quietly juggling energy, deciding whether to store it, spend it, or save it for later. This constant adjustment, this exquisite chemical diplomacy, is known as homeostasis, and it's what keeps you alive, alert, and functioning whether you're sprinting, sleeping, or simply sitting still. The body isn't static, it's a self-tuning orchestra, maintaining harmony amid constant change. At the centre of this metabolic dance lies the question of fuel. In the fed state, when you've just eaten, energy floods in. Glucose from carbohydrates, fatty acids from fats, amino acids from proteins. The body must decide where to send it all. In the fasting state, when food is scarce, it does the opposite, drawing on reserves to keep the vital systems running. Each state has its own priorities, but the goal is always the same: balance. The liver acts as the body's logistics hub, the grand coordinator of energy flow. After a meal, glucose absorbed by the intestines enters the bloodstream, raising blood sugar levels. The liver steps in like a skilled traffic controller, taking in some of that glucose and converting it into glycogen, a compact storable form of sugar. Excess carbohydrates can also be turned into fatty acids and exported to adipose tissue for longer-term storage. Meanwhile, other tissues, the muscles, brain and heart, take up glucose directly to power their work. During fasting, the roles reverse. Blood sugar begins to drop, and the liver shifts from storing to supplying. It breaks down glycogen to release glucose into the blood, keeping the brain and red blood cells fueled. Once glycogen stores run low, the liver begins making new glucose from scratch, using gluconeogenesis, a process that repurposes amino acids, glycerol, and other small molecules to rebuild sugar from non-sugar parts. At the same time, fat tissue begins to release fatty acids, which the liver and muscles use for energy. The liver can even convert some of those fatty acids into ketone bodies, which the brain can use as an alternative fuel. It's a remarkable demonstration of adaptability, every organ adjusting, every molecule finding a new route when circumstances change. Keeping this system stable requires communication, and that comes from hormones, the chemical traffic lights that signal when to stop, go, or slow down. The main trio in metabolic control are insulin, glucagon, and epinephrine, each with a distinct but complementary role. After eating, rising blood glucose triggers the pancreas to release insulin, the hormone of abundance. Insulin tells cells throughout the body that energy is plentiful. Muscle and fat cells open their glucose transporters to take up sugar, while the liver ramps up glycogen and fat synthesis. It's a time of building, of investing for the future. As the hours pass and blood sugar begins to drop, glucagon takes the stage. Also released from the pancreas, glucagon sends the opposite message. Resources are dwindling. Time to switch to internal stores. It signals the liver to break down glycogen and activate gluconeogenesis, ensuring a steady trickle of glucose into the bloodstream. Then there's epinephrine, better known as adrenaline, the hormone of urgency. When you face sudden stress or physical exertion, epinephrine overrides the normal slow hormonal rhythm. It tells the liver and muscles to release glucose immediately and mobilizes fat for quick energy. Heart rate rises, breathing deepens, and every system gears towards survival. It's the metabolic equivalent of stepping on the accelerator, brief, powerful, and perfectly timed. These hormonal signals don't operate in isolation. They interact constantly. Insulin suppresses glucagon, glucagon counters insulin, and both respond to feedback from the very changes they create. It's a tightly woven loop of checks and balances. Too much insulin for too long, and blood sugar crashes. Too little, and sugar builds up in the blood, starving cells of fuel. The precision required is astonishing, and yet most of the time the system gets it exactly right without you ever noticing. At a deeper level, metabolism is not divided neatly into carbs, fats, and proteins, but rather a single interconnected web. Each fuel can feed into another. Excess glucose can become fat. Certain amino acids can be turned into glucose. Fatty acids can generate ketones that replace glucose when needed. These pathways converge at shared intermediates, molecules like acetyl-CoA or oxaloacetate, the crossroads of metabolism. The body shifts which roads are busiest depending on diet, activity, and time of day. For example, during exercise, muscles consume glucose at high speed, pulling from both blood and glycogen stores. As exertion continues, the body gradually leans more on fats, sparing precious glucose for the brain. In contrast, during sleep, energy demand slows, and the body settles into a steady rhythm of fat oxidation. After a fast or during endurance exercise, ketone bodies step in, keeping the brain supplied when glucose is low. This seamless transition between fuels shows how flexible and self-regulating metabolism truly is. Even stress and rest represent different steady states of the same principle. Under chronic stress, hormones like cortisol raise blood sugar and encourage fat breakdown, ensuring a ready energy supply, helpful for emergencies but harmful if prolonged. In contrast, during rest, parasympathetic signals promote storage and repair, encouraging insulin's effects and replenishing glycogen. Exercise temporarily disrupts equilibrium, but the recovery period afterwards strengthens it, improving the body's ability to respond to future challenges. Homeostasis isn't about keeping things constant. It's about staying responsive without losing stability. The coordination required for all this is immense. Every tissue, muscle, fat, liver, brain, pancreas, acts like a section of an orchestra. The hormones are the conductors, keeping the rhythm steady but adjusting tempo and volume as needed. A surge of glucose here, a drop in oxygen there, and the music subtly changes. And while it might seem chaotic on the surface, the result is remarkably smooth, a continuous harmony of chemical adaptation. Even the smallest deviations are corrected automatically. If body temperature rises, blood vessels widen, sweat evaporates, and heat dissipates. If it falls, blood flow is redirected inward, and muscles shiver to generate warmth. If pH drifts, buffers and respiration adjust. Every variable, oxygen, pressure, salt, glucose, has its set range, and the body defends it tirelessly. It's not about perfection, it's about balance within limits. This principle of balance extends beyond the individual. On a global scale, life itself depends on feedback loops, ecosystems regulating oxygen, carbon, and nutrients just as a cell regulates ions and metabolites. The same logic governs both. Detect change, respond proportionally, and restore equilibrium. From the glucose level in your blood to the carbon dioxide level in the atmosphere, life thrives by keeping its chemistry within bounds. Metabolism, at its heart, is not just the conversion of molecules, it's regulated chemistry in motion. A living cell is never in equilibrium. It exists in a constant state of controlled disequilibrium, always just out of balance enough to move forward. Every heartbeat, every breath, every thought depends on this gentle instability, sustained by the endless interplay of signals and reactions. The next time you eat, exercise, or simply rest, consider the unseen adjustments taking place. Your liver reroutes carbon, your hormones negotiate priorities, your cells quietly decide how to spend their energy budget. It's an astonishing act of self-regulation, one that's been refined for billions of years. In the end, life's stability doesn't come from stillness, but from motion, from chemistry that responds, adapts, and endures. Homeostasis is not a frozen balance, but a living one, a quiet, ceaseless conversation that keeps the body, and perhaps all of nature, perfectly poised on the edge of change. Life is a balancing act. Every moment your cells are quietly juggling energy, deciding whether to store it, spend it, or save it for later. This constant adjustment, this exquisite chemical diplomacy, is known as homeostasis, and it's what keeps you alive, alert, and functioning whether you're sprinting, sleeping, or simply sitting still. The body isn't static, it's a self-tuning orchestra, maintaining harmony amid constant change. At the center of this metabolic dance lies the question of fuel. In the fed state, when you've just eaten, energy floods in. Glucose from carbohydrates, fatty acids from fats, amino acids from proteins. The body must decide where to send it all. In the fasting state, when food is scarce, it does the opposite, drawing on reserves to keep the vital systems running. Each state has its own priorities, but the goal is always the same: balance. The liver acts as the body's logistics hub, the grand coordinator of energy flow. After a meal, glucose absorbed by the intestines enters the bloodstream, raising blood sugar levels. The liver steps in like a skilled traffic controller, taking in some of that glucose and converting it into glycogen, a compact, storable form of sugar. Excess carbohydrates can also be turned into fatty acids and exported to adipose tissue for longer-term storage. Meanwhile, other tissues, the muscles, brain and heart, take up glucose directly to power their work. During fasting, the roles reverse. Blood sugar begins to drop, and the liver shifts from storing to supplying. It breaks down glycogen to release glucose into the blood, keeping the brain and red blood cells fueled. Once glycogen stores run low, the liver begins making new glucose from scratch, using gluconeogenesis, a process that repurposes amino acids, glycerol, and other small molecules to rebuild sugar from non-sugar parts. At the same time, fat tissue begins to release fatty acids, which the liver and muscles use for energy. The liver can even convert some of those fatty acids into ketone bodies, which the brain can use as an alternative fuel. It's a remarkable demonstration of adaptability, every organ adjusting, every molecule finding a new route when circumstances change. Keeping this system stable requires communication, and that comes from hormones, the chemical traffic lights that signal when to stop, go, or slow down. The main trio in metabolic control are insulin, glucagon, and epinephrine, each with a distinct but complementary role. After eating, rising blood glucose triggers the pancreas to release insulin, the hormone of abundance. Insulin tells cells throughout the body that energy is plentiful. Muscle and fat cells open their glucose transporters to take up sugar, while the liver ramps up glycogen and fat synthesis. It's a time of building, of investing for the future. As the hours pass and blood sugar begins to drop, glucagon takes the stage. Also released from the pancreas, glucagon sends the opposite message. Resources are dwindling. Time to switch to internal stores. It signals the liver to break down glycogen and activate gluconeogenesis, ensuring a steady trickle of glucose into the bloodstream. Then there's epinephrine, better known as adrenaline, the hormone of urgency. When you face sudden stress or physical exertion, epinephrine overrides the normal, slow hormonal rhythm. It tells the liver and muscles to release glucose immediately and mobilizes fat for quick energy. Heart rate rises, breathing deepens, and every system gears toward survival. It's the metabolic equivalent of stepping on the accelerator, brief, powerful, and perfectly timed. These hormonal signals don't operate in isolation, they interact constantly. Insulin suppresses glucagon, glucagon counters insulin, and both respond to feedback from the very changes they create. It's a tightly woven loop of checks and balances. Too much insulin for too long, and blood sugar crashes. Too little, and sugar builds up in the blood, starving cells of fuel. The precision required is astonishing. And yet, most of the time, the system gets it exactly right without you ever noticing. At a deeper level, metabolism is not divided neatly into carbs, fats, and proteins, but rather a single interconnected web. Each fuel can feed into another. Excess glucose can become fat. Certain amino acids can be turned into glucose. Fatty acids can generate ketones that replace glucose when needed. These pathways converge at shared intermediates, molecules like acetyl-CoA or oxaloacetate, the crossroads of metabolism. The body shifts which roads are busiest depending on diet, activity, and time of day. For example, during exercise, muscles consume glucose at high speed, pulling from both blood and glycogen stores. As exertion continues, the body gradually leans more on fats, sparing precious glucose for the brain. In contrast, during sleep, energy demand slows and the body settles into a steady rhythm of fat oxidation. After a fast or during endurance exercise, ketone bodies step in, keeping the brain supplied when glucose is low. This seamless transition between fuels shows how flexible and self-regulating metabolism truly is. Even stress and rest represent different steady states of the same principle. Under chronic stress, hormones like cortisol raise blood sugar and encourage fat breakdown, ensuring a ready energy supply, helpful for emergencies but harmful if prolonged. In contrast, during rest, parasympathetic signals promote storage and repair, encouraging insulin's effects and replenishing glycogen. Exercise temporarily disrupts equilibrium, but the recovery period afterwards strengthens it, improving the body's ability to respond to future challenges. Homeostasis isn't about keeping things constant, it's about staying responsive without losing stability. The coordination required for all this is immense. Every tissue, muscle, fat, liver, brain, pancreas, acts like a section of an orchestra. The hormones are the conductors, keeping the rhythm steady but adjusting tempo and volume as needed. A surge of glucose here, a drop in oxygen there, and the music subtly changes. And while it might seem chaotic on the surface, the result is remarkably smooth, a continuous harmony of chemical adaptation. Even the smallest deviations are corrected automatically. If body temperature rises, blood vessels widen, sweat evaporates, and heat dissipates. If it falls, blood flow is redirected inward and muscles shiver to generate warmth. If pH drifts, buffers and respiration adjust. Every variable, oxygen, pressure, salt, glucose, has its set range, and the body defends it tirelessly. It's not about perfection, it's about balance within limits. This principle of balance extends beyond the individual. On a global scale, life itself depends on feedback loops. Ecosystems regulating oxygen, carbon, and nutrients, just as a cell regulates ions and metabolites. The same logic governs both. Detect change, respond proportionally, and restore equilibrium. From the glucose level in your blood to the carbon dioxide level in the atmosphere, life thrives by keeping its chemistry within bounds. Metabolism at its heart is not just the conversion of molecules, it's regulated chemistry in motion. A living cell is never in equilibrium. It exists in a constant state of controlled disequilibrium, always just out of balance enough to move forward. Every heartbeat, every breath, every thought depends on this gentle instability, sustained by the endless interplay of signals and reactions. The next time you eat, exercise, or simply rest, consider the unseen adjustments taking place. Your liver reroutes carbon, your hormones negotiate priorities, your cells quietly decide how to spend their energy budget. It's an astonishing act of self-regulation, one that's been refined for billions of years. In the end, life stability doesn't come from stillness, but from motion, from chemistry that responds, adapts, and endures. Homeostasis is not a frozen balance, but a living one, a quiet, ceaseless conversation that keeps the body and perhaps all of nature, perfectly poised on the edge of change. Life balances itself with astonishing grace. At every moment, millions of reactions within your cells are in quiet negotiation, some storing energy, others spending it. Together, they form a stable rhythm called homeostasis, the art of keeping things steady in a changing world. This balance shifts gently depending on your state. In the fed state, the body is flush with nutrients. Glucose rises, insulin flows, and cells eagerly store energy. The fasting state is its mirror. When external fuel runs low, the body dips into reserves, rationing energy with calm precision. The liver sits at the center of it all, a biochemical air traffic controller directing glucose, fats, and amino acids to where they're needed. After a meal, it stores sugar as glycogen or converts excess carbon into fat for later. In fasting, it does the opposite, breaking down glycogen and manufacturing new glucose through gluconeogenesis, ensuring a steady supply for the brain and blood. Communication between organs comes through a handful of hormones, the body's chemical messengers. Insulin signals abundance, guiding glucose into cells and encouraging storage. Glucagon does the opposite, calling on reserves when sugar runs low. Epinephrine, or adrenaline, overrides both in moments of stress, mobilizing every resource for immediate use. They function like elegant traffic lights, green for storage, red for release, amber for urgency, keeping metabolism smooth even as conditions shift. Underneath these signals, metabolism hums as a single connected web. Carbohydrates, fats, and proteins aren't isolated fuels. They're threads of one tapestry. Carbon atoms from sugars can become fat, amino acids can turn into glucose, and fatty acids can be repurposed into ketones when carbohydrates run scarce. The body constantly balances these pathways, rerouting energy through shared intersections like acetyl-CoA, pyruvate, and oxaloacetate. Different states call for different blends of fuels. During a sprint, muscles burn glucose at breakneck speed, while endurance exercise leans more on fat. During sleep, metabolism slows and the body restores balance, oxidizing fat quietly through the night. In fasting, the liver produces ketone bodies, providing the brain with a calm, alternative energy source. Even these shifts follow the same rules: never waste, never stop, always balance. Hormones act as conductors in this metabolic orchestra, but the music itself changes with context. Cortisol rises under long-term stress, releasing glucose and breaking down fat to sustain alertness. At rest, parasympathetic signals restore calm, favoring repair and storage. The transitions are seamless, a living rhythm of ebb and flow, where stability is achieved not by stillness but by responsiveness. Every layer of this system is governed by feedback. Rising glucose triggers insulin, which lowers glucose, which in turn slows insulin release. The same logic applies to body temperature, blood pressure, and pH. Gentle oscillations around perfect balance. It's a masterpiece of self-regulation. Even the crosstalk between pathways mirrors ecological balance on a global scale. Inside a cell, carbon and energy flow like nutrients in an ecosystem. Each reaction feeds the next, waste from one process becoming fuel for another. Nothing stands alone, and nothing is wasted. Homeostasis is therefore not a static condition, but a dynamic one. It's the controlled tension between input and output, order and disorder. Cells constantly push against equilibrium to stay alive, burning energy just to maintain that poised imbalance. In chemistry, equilibrium means stasis. In biology, it means death. Life exists only in motion. This principle scales outward. An athlete mid-run, a plant under sunlight, a forest exchanging carbon with the sky. All are governed by feedback and adjustment. The same patterns that stabilize your blood sugar also help regulate Earth's climate. Whether in a single cell or an entire biosphere, life survives through balance amid flux. So when you eat, or rest, or breathe deeply before sleep, imagine the quiet choreography within. Glucose flowing, enzymes adjusting, hormones whispering instructions. Your body's not a machine that runs. It's a conversation that never ends. Homeostasis is chemistry made mindful, a living equilibrium that bends but never breaks, adapting gracefully to a changing world. It is life's oldest secret, stability born not from stillness, but from perpetual, intelligent motion. In the soft hush at the end of this journey, 3 quiet threads keep shimmering through the story. First, structure to function. The way a molecule is shaped guides what it can do. A fold in a protein opens a pocket where reactions happen. A kink in a fatty acid loosens a membrane. A spiral of nucleotides stores a pattern the cell can read. Form gives rise to purpose, again and again. From the curl of an alpha helix to the curve of a membrane budding into a vesicle. Second, energy flow. Life rides gentle gradients, letting electrons fall and protons drift, catching their motion in small spendable coins. It is the patient art of coupling what must be built to what is ready to fall. Third, regulated networks. No reaction acts alone. Pathways sense, respond, and ease one another forward or quietly apply the brakes. The result is not chaos but choreography, 1,000 tiny adjustments that make the whole feel simple. If there is a minimalist toolkit behind all of this, it might be water, membranes, enzymes, and information. Water is the stage and the lighting, invisible but decisive, pulling charged things into solution and nudging oily things together. It cushions heat, splits to offer or accept protons, and whispers to proteins where to hide their hydrophobic faces and where to show them. Membranes draw the line between inside and out, but never as a wall. They ripple and flex, gather rafts for conversation, curve into packets for travel. Across them, gradients store potential the way a hillside stores the promise of a river. Enzymes are the polite speeders of the cell, lowering hills no one else can climb, choosing the right collision out of countless wrong ones, and doing it all without fuss. And information, written in the letters of nucleic acids, is the soft voice that says what to make, when to make it, and where it belongs. It does not shout. It is read, translated, and put to work, then folded back into silence. Hold those 4 pieces together and a pattern emerges. Water sets the tone, membranes give a shaped difference, enzymes negotiate the rates, information supplies memory and plan. With just these elements, cells write the music of metabolism. Sugars gently split, carbon circled and released, electrons handed along from one careful grasp to the next. ATP appears and disappears, not with fireworks, but with a nod. Signals ring at the surface, bloom inside as second messengers, and then fade, as if someone has dimmed the lights to end the party. Oxygen waits at the end of a chain and becomes water again. Carbon enters as air and leaves as breath. The chemistry completes its loops and starts over. What is most striking is how much happens without fanfare. In every moment there are billions of tiny decisions. A gate in a membrane opens for a millisecond and closes. A polymerase backs up to fix a mismatch. A kinase adds a phosphate and a phosphatase takes one away. A motor turns a few degrees and pauses. From far away it seems like stillness. Up close, it is steady motion carried out with exquisite restraint. This is the paradox at the heart of biochemistry. Nothing is truly at rest, and yet the experience is one of calm. The pattern holds as the parts renew. If you listen closely, you can almost hear the themes repeating. Structure to function, A change in shape releases oxygen when a tissue's hungry, or tightens a channel when ions have had enough. Energy flow. Gradients built one ion at a time pay for work in careful installments. Regulated networks. A product loops back to quiet the first step that made it. A wave of calcium rises and falls like a breath. A messenger comes and goes before the cell is tempted to overreact. Even errors are part of the design. A rare mutation slips through and is mended, or now and then becomes a new note in the song of life. Control is not rigidity. It is the confidence to bend, and so the body remains a friendly contradiction. It is robust because it is soft. It is precise because it is flexible. It is predictable because it is full of options. With water to host the dance, membranes to draw graceful edges, enzymes to keep time, and information to remember the steps, the chemistry keeps watch whether you're noticing or not. It cooks and cleans the cell, changes the linens of a membrane, sweeps the pathways, and turns the lights down low. When food arrives, it stores a little extra in quiet places. When fuel runs short, it unlocks the pantry and shares. When a signal rings at the door, it answers. When the message has been received, it says thank you and tidies up. You do not have to hold any of this in mind for it to do its work. The solace of biology is that its patience does not depend on your attention. You can let your thoughts loosen, let your breathing slow, and allow the day to set. The molecules will keep their gentle appointments. The gradients will lean just enough. The folds will hold. The codes will be read and put away. The quiet will deepen, not because the chemistry has stopped, but because it is so perfectly content in motion. And in that quiet, if you feel sleep reaching for you, you can let it. The story will continue without you for a while, faithful and unhurried, and when you wake it will still be there, the same small miracles doing what they always do. Sleep well, your cells know what to do,